Alright, so this is a pretty new thing I'm going to try to do. I finally finished editing the entire PDF, right? So my goal now is to try to get through the entire PDF in this month, and instead of doing 1 to 2 hour long videos, I'm going to try to do the entire thing in one video. So I'm going to try to do like full chapters in one video, and this is pretty experimental right now. I don't know if I'm going to die by the time I'm done with this today, but I had to let everyone know that I'm not going to be able to really help anyone with anything, like family or whoever might need me possibly, because phone's going to be quiet. Because I'm going to be doing this all day, right? So it's 12 here right now, and by the time I'm done, if I even finish all of biochemistry, then it'll probably be around 6 or 7. So I know it's going to take a lot of time. Now with biochem, the majority of the bulk of biochemistry is in nutrition and metabolism, right? Those are the significantly larger sections of the book. So I definitely will be able to breeze through molecular, cellular, and lab techniques, and there's a little to talk about in genetics, but most of it is just about genetics questions, which I know a lot of people do struggle with. Now, another thing that I want to point out to you guys is, as I'm going to be going through these various chapters, right, as I'm going to be going through these various different parts of the first aid book, I'm also going to be taking note of things I'm going to need on the day of my test, right? So let's say, for example, if I'm taking my test, let's just say, for example, I'm taking my test in December then, right? So if I'm doing that, I want to have these things ready, because these are going to be the things that I'm going to be memorizing and then rewriting on my 2 pages of scrap paper that I'll have on the day of my test. Right, so I have my little portfolio thing here where I'm just gonna write things down as I'm gonna keep going, and by the time I'm done, I'll share that. I'll post it on the YouTube page so that you can see what specific notes I was able to put aside. Because those are going to be the things I'll go ahead and ultimately utilize the day, the week before my test. The week before my test, I'm going to go ahead and organize my cheat sheet, which is going to be basically something I'll keep rewriting by hand on my own paper until the day of my test, right? And then when the day of my test comes, I will go ahead and be able to reproduce that on the 2 pieces of paper that I will get. Thank you, Basima. Eid Mubarak to everyone that celebrates. And yeah, I can email it to anyone who needs it. I can also email the PDF to anyone who has already purchased it but doesn't have the most updated version. So I've finished it now. There's really nothing more to add. I mean, in terms of questions and stuff, at least I'm done with everything I could possibly add to the PDF. So it's pretty much completed. Not pretty much, it's completed. I've added everything. The only thing I could possibly ever add anymore is going to be something like brand new that I might find somewhere. But other than that, it's done. I finished everything all the way to respiratory, added a bunch of AMBOSS notes and everything. But yeah, so this is pretty critical, because it's one of the most important things that I felt like I needed to do when I took my step 1, right? So when I sat down for my step 1 and they gave me these 2, if you haven't seen, the paper that you get is actually a plastic kind of thing. You can smudge it, you can't really erase it per se, but they can erase it. So, and it's double-sided, so you're essentially getting 4 pieces of paper. You're getting 4 pages, if anything. You're getting 4 pages. So on those 4 pages, you can write down whatever you want. Different people prepare different ways for it. The way I prepared for those 4 pages was that I was ready to reproduce things that I need to memorize for temporary, tiny, easy questions. Right, but those easy questions become ridiculously difficult if you did not memorize what you needed to know. Now, in biochemistry, there isn't much in terms of things that need to go on these pieces of paper. But when you get to genetics, you'll see equations there. So there's that, and then metabolism is one of those chapters where metabolism you just have to look at like. Every time you do one of these chapters, you finish cardiovascular, go back and look at metabolism. Then you do all of endocrine, go back and look at metabolism. You finish renal, go back and... You need to revisit metabolism over and over, because it's really entirely just a memorization game. The more times you talk about it, the better you get at it. Right? I also won't necessarily be muting my phone because since I'm doing this for so long, people will definitely need to call me at some point, so I will be answering that. But other than that, that's it. All right, so we gave 5 minutes for people to join in, and they should be. Let me see. Yeah, it's, all right, 12 people. Great. All right, so we're gonna get started. I'm gonna start now. I'm gonna keep going through all of biochemistry until I cannot talk anymore. I definitely won't get hungry, but you know, if I do need to walk away for anything, I'll gladly just go ahead and do that. So let's get this going. So biochemistry. All right. So in the beginning, you know, these things are pretty easy, pretty straightforward to breeze through. But easy doesn't mean, you know, that you're gonna get it right on the test. So you got to memorize these things. Chromatin structure. Right. This is a great picture. Pretty unique picture. Not many other books have a picture like this. DNA exists in the condensed form, which you see all the way over here. Chromatin formed to fit into the nucleus. DNA loops twice around the histone octamer to form a nucleosome. So right here you can stop. Now you got to think, you know, one mistake many people make when they're studying and reading stuff is that they read something and they take for granted that they perfectly understood what they read. So you got to make sure you stop and really do understand what you read, right? So there's this DNA looping twice around the histone octamer is something you definitely don't want to get wrong if you do get a silly, stupid little question on that, right? So that's what you see in this little visualization I put over here, right? You can see it went around twice on an octamer, 8 different histone balls that are there. They all have their unique names, H2A, H2B, H3, and H4. There's 2 of each, right? And how they're organized is not something you need to know. You just got to remember that you see even here they show you, right? 2 loops around the entire 8-ball thing. And then you can also see there's this one other guy here known as an H1 histone linker. Right, so that's the next thing they talk about. H1 binds to the nucleosome and to the linker DNA, thereby stabilizing the chromatin fiber. All right, so if you don't have the H1 linker, these loops, this double loop that you have around the octamer can get messed up and it can open up. Phosphate groups give DNA a negative charge. Lysine and arginine give histones a positive charge, right? And you're going to see more about this, I think, later on in molecular, where they talk about basic and acidic and positive and negative charge or neutral amino acids, right? So lysine and arginine are positive. Phosphate is a negative one. We all know about phosphate very well. ATP has phosphates on it, right? Phosphate groups carry a negative charge. So this negative and positive charge is what kind of keeps these guys linked together as well, right? Very important. So one little thing I had made up was histones are large, which would help me remember lysine and arginine, and it just stuck to me. That was something I personally made up myself, and one of the major reasons I did actually remember that was because I made it up. In mitosis, DNA condenses to form chromosomes. DNA and histone synthesis occurs during S phase. And we're going to see more about this when we get further on into cellular. Right? But mitosis. So mitosis and meiosis are big deals. We're going to talk more about that in reproductive as well. But just remember that DNA is condensed to form chromosomes, and DNA and histone synthesis, don't forget the histone part, both things are synthesized during the S phase, which is pretty much the synthesis phase. Mitochondria have their own DNA, which is circular and does not utilize histones. It also has many different exceptions that we're going to see as we move on. And so over here I wrote the lysines on histones are particularly important because gene transcription is often regulated by covalently adding or removing methyl or acetyl groups on these lysines. Right? We've all heard of methylation and acetylation, where methylation mutes things and acetylation activates things, activates transcription for the most part. So that methylation and acetylation is usually done using the lysine. All right. And based off of that, either your DNA is going to be too condensed, so it can't be transcribed, or it'll be very widely open and it can be transcribed by enzymes accessing it. These are just a vocabulary list of things here, right? So heterochromatin and euchromatin are pretty straightforward. There's nothing much there. DNA methylation, and then the last 3 are about histone methylation, acetylation, and this is relatively new thing in first aid 2019 about histone deacetylation. So DNA methylation mutes things, right? And they have that here written. Methylation makes DNA mute. DNA methylation changes the expression of a DNA segment without changing the sequence, right? You're just doing what we just talked about, methylating a lysine residue possibly. And what that does is it kind of takes your DNA, and you see how methylation kind of made all the DNA just become really smushed together. So enzymes can no longer access different parts of this anymore to do transcription. Whereas acetylation up here, you can see is doing a different thing. Now these images over here are for histone, right? What we're talking about up here is DNA methylation. Though DNA methylation is something that happens to genomic imprinting, X chromosome inactivation, which is the bar body, repression of transposable elements, and in aging, very common thing in aging, and in carcinogenesis, right? So these are all different scenarios where you have DNA methylation. And if DNA is methylated in imprinting, then you're going to have all those imprinting diseases that we're going to talk about under genetics. Methylation within a gene promoter, which is a CpG island, typically represses the gene transcription, right? If you mute the promoter, you're no longer going to be doing that promoting, and then you're going to slow down and possibly even silence the transcription of certain genes. The P in between just represents the phosphate back chain, right? That's how these guys are all kind of linked. Now, for histones, you see this image here, right? So these are the histone octamers. You can see the double looping around it, right? If you have histone methylation, you cause all of it to just stick together like this, right? Reversible transcriptional suppression, but can also cause activation depending on location of methyl groups. Now, histone acetylation, removal of histone's positive charge, leads to relaxed DNA coiling, right? Increased transcription. So histone acetylation is actually removing the positive charge from histones, which basically would be, you know, the lysine and arginine are the guys that give that positive charge. And when you do acetylation, you just get rid of that positive charge, so all of the negative charge starts repelling each other, and you can see how they've all kind of gone further apart from each other. Alright, so that's histone acetylation. Histone acetylation makes DNA active. And this little note here came from an NBME question that a lot of people had gotten really upset about, where in this question they asked how thyroid hormone actually exerted its final effect. And I googled it and I looked up a bunch of stuff because people mentioned methylation, acetylation, and stuff like that. So thyroid hormone actually works via acetylation. Alright, so that was one of the things that I wanted to make sure I put in over here. Then you have histone deacetylation, which is pretty much akin to histone methylation, but not exactly, right? Histone methylation made it really, really tight. Histone deacetylation will just undo this, you know, distancing of histones. So you remove the acetyl groups and you have tightened DNA coiling and decreased transcription. Decreased transcription, very similar to how histone methylation would cause decreased transcription, but not exactly the same thing. Now, over here on the right side, you see images of nucleotides, which are coming up next. Nucleotides, you don't necessarily need to know how they look per se. You don't have to have perfect memory of how they look, like, you know, the way I've written it here. But you do need to understand that purines are the ones that have 2 rings, and pyrimidines have 1 ring, right? So pyrimidines I always think of as a pi, right? That's what I wrote here. So pyrimidines, because you do have that the mnemonic cut the pi, right? So CUT, cytosine, uracil, and thymine are purines. And then it's also the one that looks like a pi, which means that it's literally just 1 ring, okay? The purines have 2 rings, and that's something that you can always, you know, get a question on where they just mention something is 2 rings or 1 ring, and you got to be able to answer that. Yeah, the redo videos are pretty much exactly what I'm kind of redoing right now, so those redo videos are good enough, but in this final version now, I have added like a question or 2 here or there, so I want to make sure I get everything online before I move on to CK videos. Alright, so nucleotides, you see this image here, you kind of get an understanding of the definition of what exactly is a nucleotide. Right, so you see the yellow portion, this yellow portion with the pentose phosphate, you have your ribose and your deoxyribose, depending on whether, you know, ribose is going to be on RNA, deoxyribose is on DNA, and you have your glycosidic bond with your base. This yellow portion is a nucleoside. As soon as you add 1 phosphate, now you have a nucleotide, all right? But then when you start adding more phosphates, that's also known as just nucleosides, all right? The only thing that's a nucleotide is a base plus a deoxyribose plus a phosphate. That's it. right? Or a ribose. It doesn't have to be a deoxyribose, deoxy or just a normal ribose. But point is, just one phosphate is what makes a nucleotide, right? As soon as you have more phosphates or no phosphates at all, then that's a nucleoside, right? And that's very important to remember. So that's like a little definition kind of thing that you can get a silly question on. Other than that, just basic things here about, pure as gold, cut the pie, just to memorize things. And thymine has a metal. That's something that you can get a question on where, you see right here, this is the metal group that's sticking out right here from organic chemistry, if you remember that notation. No one else has a metal. Right, so if you ever get a question on one of these nucleotides and they tell you that there's a metal there, thymine is the only one that has a metal. So they must be talking about thymine. The GC bond has 3 H bonds, and it's stronger than the AT bond, which has 2 H bonds. So a higher GC content means you have a higher melting temperature. It's going to take more heat to separate that bond, right? It also, then I had another question on the same kind of topic, so I made this note where I wrote DNA length also affects melting temperature because longer DNA requires a higher melting temperature, right? So not just how much GC content you have, but also the length of the DNA. And of course, length of the DNA does kind of fall into the same category as how many GCs do you have, right? So, you can get a silly question on that. Just remember, GC is a very strong bond compared to the AT bond. Amino acids necessary for purine synthesis. They have this amount of cats per until they gag. Right? So the per part is for purines. The gag part is for glycine, aspartate, and glutamine. And you do have to remember these guys. Right? So glycine, aspartate, and glutamine. Glycine and glutamine come up a lot later on when we start talking about hyperammonemia. Right? There's going to be a lot of things happening with glycine and glutamine back there. Then the deamination reactions are right here. Now, deamination reactions, you're not going to memorize the first time you look at it, but it's not the hardest part either, especially when we get to this table here, you start seeing it even more, right? So deamination of cytosine gives you uracil, adenine becomes hypoxanthine, guanine becomes xanthine, right? And then there's that HGPRT enzyme that kind of has these names in there too. And then 5-methylcytosine becomes thymine. Now, this one is really easy to remember because remember, thymine was the only one that has a metal. So 5-methylcytosine gives you thymine. Now, guanine and xanthine, and then adenine and hypoxanthine, that's up to you how you want to memorize it. And cytosine becoming uracil, I just remember that because they're in the same place, right? Cut, C-U-T. So cytosine becomes uracil. You can see, just by looking at it, that they're pretty much the same exact thing except on the top. Right, you deaminate, right? You take and you do the deamination reaction up here, you get oxygen instead, and then you have uracil, all right? So that's it for that. What I wrote here is that I'm actually writing out what you're getting. That's kind of what they're showing you here, right? The glycine gave you your CCN up here. Aspartate just gives you that 1 nitrogen. Glutamine gives you 2 nitrogens, and they point that out here as well. THF gives you 2 carbons, and CO2 gives you a carbon, and that's also kind of pointed out as well. All right, so you can see your 2 carbons that came from THF, and then your 1 carbon that came from CO2. All of those are actually things you need to make a purine, but here they only listed out the amino acids that you need for making a purine, right? THF and CO2 are not amino acids. So I just added them because those are also things that you do need to make an amino acid. Whereas to make a purine, making a pyrimidine really just takes carbonyl phosphate and aspartate. That's it. Just those 2 guys. And we're going to see that on this next page now, right? So this next page kind of talks about de novo pyrimidine and purine synthesis. Man. Alright, so various immunosuppressive, anti-neoplastic, and antibiotic drugs function by interfering with nucleotide synthesis. Now, these medications tend to actually show up a lot, especially on, well, actually, I don't really remember UWorld so much anymore, but when I did the AMBOSS questions with a bunch of, with 2 students I was working with, I did see leflunomide show up once, and then I saw questions on 5-fluorouracil and 6-mercaptopurine all the time. Right, it's a pretty common thing, and then of course hydroxyurea is a very common medication. But remember, step 1 is trying to lean away from picking on things that are very common. So you can get questions on mycophenolate, you can get questions on ribavirin. It could literally be anything. All right. So just make sure you know these guys, and for the most part, it's really just this is just an introduction. For the most part, you're gonna see these guys again. Alright, so let's look over here what's happening. So we talked about what you need to make a purine, right? That was the PERT and GAC thing right here. You need glycine, aspartate, glutamine, and then THF and CO2 add things. So making the purine was the easier part, right? Purine-based production, you take your ribose 5-phosphate all the way up here, and keep in mind this ribose 5-phosphate. Later on, we're going to see where this ribose 5-phosphate came from. It's going to come from the pentose phosphate pathway, all right? So ribose 5-phosphate right here, PRPP, phosphoribosyl pyrophosphate synthetase, PRPP synthetase, is going to go ahead and make PRPP out of that ribose 5-phosphate. Now PRPP is kind of the starting point for both guys. So PRPP on the purine side, you just have a bunch of unnecessary reactions that you don't need to memorize happen over here, ultimately giving you one IMP, right? And that's an inosine monophosphate. And then the IMP is either going to become adenine or guanine monophosphate based off of whatever base is attached at the end. And that monophosphate, remember, having just one phosphate, that is a nucleotide. Anything else would be a nucleoside, right? So there's that. And then you can see the medications that work on this, right? So they're all medications. These guys are medications that kind of affect the purine synthesis stuff, right? And then they tell you that here, purine synthesis. So 6-mercaptopurine, that's in the name. Azothioprine is also, you can actually pronounce that as azothioprine as well. Both of those guys inhibit purine synthesis. Mycophenolate and ribavirin are two medications that you're going to read more about when we get to microbiology. They inhibit IMP dehydrogenase. Right, so if you can't do IMP dehydrogenase, you cannot make GMP. Right, so you lose guanine. That's what's happening on this side. Now over here, for the pyrimidine production, that requires aspartate and glutamine. Right? And we kind of just saw that up here as well, right? Aspartate is the big part right here. There's going to be glutamine being used, and carbonyl phosphate is going to enter later on. This is the next guy right here. Right? Glutamine and CO2 with this enzyme known as CPS2, carbonyl phosphate synthetase 2, is going to go ahead and make carbonyl phosphate. Very well named enzyme. Right, so CPS2, carbonyl phosphate synthetase, is going to make carbonyl phosphate. Then you add the aspartate right here, right? Combine it with this carbonyl phosphate, and that gives you orotic acid, which is a pretty important part right here, because you get diseases regarding this, which is known as orotic aciduria, as well as something that you can actually monitor for the most part. So this exact area is where leflunomide stops dihydroorotate dehydrogenase. Dihydroorotate dehydrogenase, right? So orotate. Orotic acid is also just known as orotate. So without that enzyme, you won't be making any orotic acid. And then later on, you just won't be making any pyrimidines either without the orotic acid. If you don't do that, then the orotic acid later becomes UMP, right? And then this part right here gets messed up in another problem known as orotic aciduria, which we're going to see much more about as we get to the end. But just remember that since it's right here, right, the major issue in orotic aciduria is the lack of UMP. Right? You're not going to have uridine monophosphate. That is the big problem in orotic aciduria. So all you really need to do for those patients is give them UMP. Right? Once they have the UMP they need, they can combine PRPP and UMP to make UDP, which is now a nucleoside, right? It's got 2 phosphates on it. And then you have your ribonucleotide reductase, which is the target of hydroxyurea. It stops it. If you have hydroxyurea as a medication you're taking, you're no longer going to be using the UDP either. But without that aspect, you continue going. CTP is very easy to make, but the thiamine is the hardest one to make. Right, DTMP right here. There's many ways to stop it. You have your 5-fluorouracil, capecitabine, methotrexate, TMP, and pyrimethamine. All of these medications that they kind of mention over here, and you use them as depending on whatever the patient needs, because it works in humans, bacteria, protozoa, respectively. So if someone has a protozoal infection, you just give them pyrimethamine, and you won't affect the human because everyone has a different thing. But ultimately, those medications stop dihydrofolate reductase, which goes ahead and recycles THF Without THF, you can't really make DTMP. Alright, so notice how in the end these are all basically MPs, AMP, GMP, TMP. The only one that's not is the CTP, right? It's the CTP triphosphate. So that one is very different from the others. So it's a little thing that many people tend to overlook. Just make sure you don't overlook that. Over here they added this thing telling you that there are two CPS enzymes. There's CPS1, which is in the mitochondria, and CPS2, which is in the cytosol. This was CPS2 right here. CPS2 was the one that was needed to make pyrimidines. CPS1 in the mitochondria is going to be part of the urea cycle, and we're going to see more about that later on. And so I just spoke about all of this that's noted up here. These two images are related to what we just saw as well. Right? So the de novo purine synthesis. You had your ribose 5-phosphate. You made PRPP using PRPP synthetase. The enzyme that first aid doesn't talk about is PRPP aminotransferase, which actually takes your PRPP and makes 5-phosphoribosylamine. This 5-phosphoribosylamine is then made into IMP, which goes to GMP or AMP. Right? The things that are happening over here. So notice how it skipped a bunch of steps over here, right? So I kind of just needed to include this one step because PRPP aminotransferase is actually a regulatory enzyme for purine synthesis, because you can see that it's inhibited by the end products, right? If you have a lot of AMP, a lot of GMP, or a lot of IMP, then it will go back and do negative feedback on PRP aminotransferase. All right, so if you have high amounts of these guys already, you don't need to make more of it. So in that case, PRPP aminotransferase activity would be decreased. All right. Over here, we're talking about the medications we just looked at. So azathioprine, remember, azathioprine becomes 6-mercaptopurine, which is actually what it's called, it's mercaptopurine. This is a prodrug for that. And what this medication basically does is it, has an impact on multiple things. Over here, you're seeing active metabolites that come from 6-MP. Those active metabolites inhibit purine synthesis, right? To become active, it needs the enzyme HGPRT, which our body uses itself. Then there's another enzyme known as TPMT, which makes inactive metabolites. It breaks down 6-mercaptopurine. All right. Over here, they wrote xanthine oxidase, which is a medication, which is an enzyme mostly. It's an enzyme that works on the same guys as well. And if you have allopurinol, if you're taking allopurinol is a medication that stops xanthine oxidase from working, right? You would give it to a patient with gout or something, high amounts of uric acid. So if you do have a patient who's taking allopurinol and you're inhibiting their xanthine oxidase, then notice that means you cannot break down 6-MP. You can't inactivate it. So you're going to have higher amounts of 6-MP in the patient. So if you have a patient who's taking allopurinol, you give them a lower dose of 6-mercaptopurine, right? Just lower the dose because it's going to be around longer. You're not going to be able to inactivate it as quickly as you would without the allopurinol. Now, this is your purine salvage, right? So if you have deficiencies in salvage, then you end up wasting a lot of it. Salvage basically means that as you're coming from up here to down here and you're about to urinate it, you salvage. Salvage kind of means like saving it. You recycle it, right? So if you do not recycle it, if you don't send it back upwards, then ultimately you will pee it. Right? But this isn't exactly a great thing. It's kind of like wasting things that you could recycle, right? And recycling is a great thing, so you don't want to waste things that you could possibly recycle. So you see here, GMP, IMP, and AMP. Those are the main three that we were looking at for purine synthesis, right? Pyrimidine salvage is not really an important thing, so that's not a thing that you're looking at. It's really just a question about purine salvage. So we saw the top portion, and we saw this whole thing coming downwards. You see the nucleosides are guanosine, inosine, and adenosine. Then you have your free bases, which are guanine, hypoxanthine, and adenine. And remember, these are the deamination reactions, right? Your adenosine giving you hypoxanthine, and your guanosine giving you your xanthine, right? Those are the deamination reactions we saw earlier, where cytosine became uracil and methylcytosine became thymine. So we don't care about the C and T pyrimidine guys right now, but here we do care about purines. So this kind of further helps you memorize the deamination reactions. Now, there's these green enzymes, right? So you see ADA and HGPRT, as well as APRT and RASP uricase and xanthine oxidase. Those are all your green enzymes that you need to perfectly memorize because you can get easily any questions on these guys. Now, what's happening with salvage? So to salvage it, from hypoxanthine and from guanine, you can return back to GMP and IMP using HGPRT. That's the enzyme. If you don't have this enzyme, hypoxanthine, guanine, purine, blah, 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 where is the full name for it? HGPRT. Oh, didn't even write the full name in the book? Oh, right here. Yeah, hypoxanthine guanine phosphorybosyltransferase, right? That's what the PRT means. So if you don't have this enzyme, then you're not going to recycle these two guys, and you're just going to end up making xanthine ultimately, and then you're just going to pee it out. But you notice this isn't exactly a good thing, because if you're doing this, if you're not salvaging it, you're going to have high amounts of uric acid, and high amounts of uric acid you're going to see can lead to issues where you have your the patients with gout. Okay. It mentions low dose aspirin. But what exactly do you mean about the aspirin here? So yeah, tell me what you're trying to point out, and I'll definitely talk about it. Yeah, so if you don't do this, then you're going to lose those guys. And then over here, what they're showing you is adenine is actually salvaged using another enzyme known as APRT, adenine phosphorybosyltransferase, right? Adenine has its own salvage thing, all right? So you got to remember that. They all have their own salvage things. And then now they talk about what happens when you have deficiencies of those enzymes. If you have adenosine deaminase deficiency, right, ADA, this is a different one right here. This is the one that does the deamination reaction of adenosine ultimately becoming hypoxanthine. If you have a deficiency of ADA, then you're gonna have SCID. All right, SCID. This is only one of the many different things that actually causes SCID. So SCID can be problematic because that's, immunodeficiency, a severe combined immunodeficiency. And you're going to get questions on that. We're going to see a lot more about those immunodeficiency diseases when we get to immunology. But this is the autosomal recessive manner of getting SCID. So it's not exactly the more common way, but it's definitely one of the ways. And what basically happens is that you need ADA to degrade adenosine and deoxyadenosine. But if you don't have ADA, then you're going to have high amounts of DATP, which then leads to lymphotoxicity, right? Lymphotoxicity, and that's not a good thing. You don't want lymphotoxicity. Ehlers-Danlos syndrome is a pretty rarely tested thing now because it's just so straightforward. I would still say it's pretty high yield. Excuse me. And UWorld did find a very good way to ask a question about this that people would actually get wrong. And that's where this image kind of came from, the de novo purine synthesis, because it was about the PRPP aminotransferase. So what you see here is without HGPRT, you will no longer be able to convert, you're not going to be able to go back this way with hypoxanthine to IMP or guanine to GMP. So you're going to lose all of that, right? Results in excess uric acid production and de novo purine synthesis. You're going to lose your IMP, you're going to lose your GMP, not exactly losing your AMP per se, right? AMP has this, a different thing happening there, so don't worry about that. Remember, the body only really needs to salvage GMP and IMP because IMP can give you more AMP. So very rarely is a question about AMP. So you lose your GMP and IMP, and then that's going to lead to hyperuricemia. But then you get these other effects that occur as well. You're going to get gout, which is because of hyperuricemia. But one of the more unique things is this aggression and self-mutilation thing that happens. And the other unique thing is this: the hyperuricemia is so bad that it eventually leads to orange sand in diaper. Right? You're going to see these sodium urate crystals. And of course, you can also get dystonia, which is kind of related to hyperammonemia in a sense. So they had a question about, what activity has to increase in a patient with HGPRT, right? So in a patient with HGPRT, and they didn't have PRPV synthetase as an answer. So if you have HGPRT deficiency, if you have Ehlers-Danlos syndrome, then you're not going to have these guys. You're not going to have GMP, you're not going to have IMP. So then in that case, PRP aminotransferase activity and PRP synthetase activity both have to increase. Right? When their activity increases, you make more of these guys and you replace the GMP and IMP that you lost. So they introduced PRP aminotransferase in UWorld to really bring your attention to a couple of those skipped steps. Now, the thing about aspirin, yes, aspirin is a very dose-dependent medication, right? So you can give low doses, moderate doses, or high-dose aspirin, depending on what exactly it is you're trying to achieve. And when we talk about aspirin later on, we'll definitely see more of it. But yes, aspirin is related to stopping this enzyme known as rasburicase, which kind of breaks down uric acid into urine. And rasburicase is a good enzyme. That's the only enzyme that's really getting rid of your uric acid. So if you don't have rasburicase, you're going to get gout. Okay? Probenecid upregulates rasburicase, and that's a good thing for these patients. Xanthine oxidase is the enzyme that's actually breaking down these guys and making more uric acid. So you kind of want to stop xanthine oxidase activity. You don't want to break these guys down and keep making more and more and more uric acid. That's not exactly going to be beneficial to a patient who's already suffering or is about to suffer with gout. Okay. Alright, so there's that. Genetic code features. These are pretty straightforward, more vocabulary things, right? So it's unambiguous, which means there's nothing ambiguous about any codon you see. So if you see a codon, you know exactly what it makes. So if you ever see CCC, you know it's going to make proline. There's no confusion about it, right? It's also degenerate and redundant, which means that proline can be made by many other guys, right? So there's also CCU, CCA, and CCG. Those guys will also make proline. So it is redundant, right? It's repetitive. It's degenerate, as in it's not one-to-one. There is no one-to-one function where only one codon makes one specific amino acid all the time, right? Every codon, yes, codons do specify for only one amino acid, but amino acids can be made by multiple codons, as you can see over here, right? So CCU will only ever make proline. CCU will never make anything else. That is unambiguity. But CCC, A, and G also making proline is degenerate or redundance. And the wobble position, now wobble position is a very confusing terminology for many students, right? Because the way they talk about it here, they tell you wobble codons that differ in the third wobble position may code for the same tRNA amino acid base. Specific base pairing is usually required only in the first two nucleotide positions of the mRNA codon. Now, look at this image. This is just a snippet from the same chapter just later on, right? You're looking at a tRNA, and this exactly here is your wobble position, right? So you have your 5' to 3' direction. So this is 5' CA. I can't remember what that was actually on that page. I think it was CAG or CAU. I'm not sure. Well, yeah, I think it was CAU. Either way, whatever it was, this specific spot on your tRNA is what's known as the wobble position, right? And then, of course, many people also refer to this third spot over here on your mRNA stuff to be your, sorry, not your mRNA, the third spot on your, mRNA codon to be your wobble position. And it's not necessarily the case. So these are not the wobble positions per se, right? And why is it called the wobble position? Because you can see all you're really changing is that third position on this, right? The first two guys are staying the same, but changing the third guy didn't really change the amino acid you're making. But what really decided this wobbling to occur was actually that first position on the tRNA, all right? That is the first position because you read things from 5 to 3. So depending on how your answer choice is worded, you got to be careful in picking that. Is it the biggest deal on earth? Not necessarily. It's not that big of a deal, but if you get everything right, then that's always great. Then you have your commaless or non-overlapping statement, which basically just means once you start reading, you keep reading. You don't stop. You don't go back and repeat something you read. And, there's just no pausing. You just go from left to right and read the whole thing and you make your protein. It's also universal, and there is an exception for this universality, which is your mitochondria, right? So if you do get a question where the genetic code is not being conserved, so maybe in some kind of DNA they found in some kind of experimental question you get, CCU is making something other than proline, then there's a very high chance it came from the mitochondria. That's just what you want to remember there, right? And the exception for the commonless non-overlapping part is your viruses, right? Remember, viruses are not cells. They're not living things. They're not organized. They don't require energy per se. They don't have organs. They don't have organelles. So viruses also have this. Some viruses, although I've never apparently really seen a question on this, but apparently some viruses are not commonless or non-overlapping. So they do pause, they do have overlap, they do reread portions, they go back and start from a different plot. So, that's something that you got to watch out for. So these exceptions are easily testable because why not, right? Everyone loves exceptions, and they're like one of the best things to test someone on. 1 second. All right. DNA replication. Now this page itself is just, it's something you would spend like a good 30 minutes on. Just make sure you do have these guys down pretty well, because you don't wanna get a question on these guys wrong at all, right? Because they're all very straightforward. They're all very different. One of the things that UWorld had questioned someone on, UWorld had a question on Bloom syndrome earlier. So now 2019 they've included Bloom syndrome into the PDF. But they still don't really talk about it much, right? So what does it do and whatnot? So I still kept my notes. So DNA replication. Eukaryotic DNA replication is more complex than the prokaryotic process, but uses many enzymes analogous to those listed below. Right? So this is mostly information on prokaryotic stuff. All right. In both prokaryotes and eukaryotes, DNA replication is semi-conservative, involves both continuous and discontinuous Okazaki fragments synthesis, and occurs in the 5' to 3' direction, which are all tiny, straightforward facts that you just have to remember. So a good thing to do is to make sure you're just looking at this specific image as you look at each one of these guys, right? So you have your origin of replication, particular consensus sequence in genome where DNA replication begins, maybe single or multiple. Now in eukaryotes, if you have a very long DNA, there will be multiple open spots in it. Those are all your various origins of replication, because you don't go ahead and just do replication at one part of a DNA. And it's crazy that the body is able to do this without messing up. But you start replication at multiple different parts of the DNA and then just combine it all. Somehow the body just manages to do that without making mistakes that it doesn't catch again. Of course it makes mistakes, but it does catch it. AT-rich sequences, such as the Tana box regions, are found in promoters and origins of replication. Okay, so if we come down here, you have your origin of replication. You see how the origin of replication is all the way over here? This place over here, or this place in the middle, is your origin of replication. You see the whole DNA strand, and then you have this opening in the middle. The central part is your origin of replication, and a very common mistake many students make is that they think of this as the origin of replication, but this is known as the replication fork, which is definitely a better name, right? Or the replication fork, because there's two different ways that things are going. It's like a fork in the road. So origin of replication is dead smack in the center. And that origin of replication is exactly what picks whether you're going to make the leading strand or you're going to have a lagging strand approaching the origin of replication. And that's definitely something you want to watch out for. Very technical little silly things you can get easy questions on. The replication fork is a Y-shaped region along the DNA template where leading and lagging strands are synthesized. Right, so that's this Y-shaped region down here. Your replication fork. And you can see it pretty much on both sides. This is going to keep going on both sides until you replicate the entire DNA, or you crash into another replication fork that was made from another origin replication, and you just join everything and you have your replication done. Alright, so that's your replication fork. Helicase unwinds DNA template at the replication fork. Alright, so helicase is this blue guy in this animation. Now this link doesn't work anymore. So either the guy kind of like lost the page or he got his video taken down. But this used to be a very nice video on Facebook that I used to use before. But I do encourage you to go ahead and find another video and just use that. I actually have continuously been forgetting to do that, so I haven't had a chance to put a video link here. But I'll go ahead and do that when I find it. Or if you find it, just go ahead and share that with me. But it's nice to be able to visualize this activity in action. Right. So what is helicase doing? Helicase basically, it's you kind of see this looks like a zipper, right? So this is kind of unzipping it. It's literally what you would hold to unzip any kind of zipper. So helicase is your zipper enzyme in a sense, and it unwinds DNA template at the replication fork. Helicase halves DNA, and if you have a deficiency of helicase, you get Bloom syndrome. Bloom syndrome is an autosomal recessive syndrome where you have growth retardation, immunodeficiency, and a photosensitive rash. Now, growth retardation and immunodeficiency are pretty non-specific, but when you have this group of syndromes where you have your growth retardation, immunodeficiency, and a rash, those three things together are really only seen in helicase, all right, in helicase deficiency. So there's that. Single-stranded binding proteins, or SSBPs, they prevent the strands from reannealing. Now, if you don't know what that, why that's important, where's a D? So there, these little 4 guys and 3 guys over here. You see those little hills that you're seeing? If you actually go ahead and take 2 strings, right, and you kind of wrap them like this DNA, and then you try to like bring your finger down the middle and unwind it, you'll notice that as you bring your finger down, this portion right here gets tighter and tighter and tighter, and it's just not gonna really work really well. So that's one thing. And then of course, as you're bringing your finger down on the top, they're gonna stick to each other again, right? As this replication fork keeps progressing, what's to keep this portion and this portion apart from each other? Right? Not much. I mean, until the leading and lagging strand don't arrive, these 2 guys can reanneal. Or right over here, these 2 guys, as helicase moves down, these 2 guys can then combine again. So single-stranded binding proteins, their job is to make sure that this once helicase opens it, these 2 guys do not combine. Right? So the way this is named, I always think it as single-stranded. This guy's job is to keep something single-stranded. Right? It does not want them to combine and become double-stranded. DNA topoisomerases, they take care of the other issue I've mentioned, right? They create a single or double-stranded break in the helix to add or remove supercoils, right? That's exactly what's happening down here. As helicase is moving down, the DNA is becoming really, really tight at the other end, right? And that's just something that happens. It's a physics kind of thing. So topoisomerases will continuously keep making little breaks in between to make sure there's no supercoiling, because if you have supercoiling, helicase will no longer be able to keep going, and that DNA can break and everything can just go bad. All right, so those 2 guys are just there for functional reasons of making sure DNA is kept well. Replication is bidirectional in prokaryotic or eukaryotic. It's bidirectional in both. It's bidirectional in both. This image here is given for a prokaryotic thing that's happening, but this is the same thing that happens in eukaryotes as well. Leading and lagging strand, and it's bidirectional. It's happening in both directions. But both of those directions follow the same thing though. There's really only one direction, which is your 5' to 3' direction, right? But bidirectional because you're going left and right, and you're replicating everything. But both of those, all these arrows are pointing in the 5 to 3 direction. So that's your DNA topoisomerases. Now there are medications that can stop topoisomerases from working. And that's great, right? Because if you stop the topoisomerases from working, then you will have supercoils and DNA will break, thereby disrupting DNA replication. Right? If you can disrupt DNA replication, then you can kind of work on cancer. You can work on targeting any cells that are tumors and stuff like that. So they tell you here that in eukaryotes, irinotecan and topotecan inhibit topoisomerase 1, and etoposide and teniposide inhibit topoisomerase 2. Right, so I used to have a note written here, but I guess it kind of went away. Yeah, there's no note. Well, I'll tell you what it was. Basically, the way I memorized these medications was, for this first one, the one that inhibits topoisomerase 1, I remember it as I can, or I know I can, whatever you want to use, but I can, right? This I can, the I is supposed to be for this I, which is your Roman numeral 1. Irinotecan and topotecan, the can guys stop the I can, so topoisomerase 1. And the etoposide and teniposide stop topoisomerase 2, so there's two sides to everything, right? So that's how I would remember the etoposide and teniposide medications. And that was way in the beginning when I was first studying. Later on, as you keep going, it just becomes easier and easier. So, but in the beginning, that did definitely help me a lot. In prokaryotes, fluoroquinolones, which you're going to see a lot of in microbiology. And remember, I'm going to tell you this over and over so you can get this, into your mind. But microbiology uses a lot of visual annotations, right? So the visual annotation for fluoroquinolones is anytime you see someone holding a flower in Sketchy Micro, it means that you could use fluoroquinolones to kind of treat this issue, right? So fluoroquinolones, or 4-oquinolones, inhibit topoisomerase 2 and topoisomerase 4. Alright, so that's why I call it 4-oquinolones. And, it's just a math thing, right? 4 and 2, they're both heavily related. 2 times 2 is 4. So 4-oquinolones stop topoisomerase 2 and 4. Topoisomerase 2 is also known as DNA gyrase, right? And I hate when things have multiple names, as you guys already know, but it has multiple names. So you can get a question on fluoroquinolone mechanism of action, and the answer would be DNA gyrase and not topoisomerase 2. Then you have to make sure you remember DNA gyrase is topoisomerase 2. Primase basically just makes an RNA primer on which the DNA polymerase 3 can initiate replication. You need a topoisomerase 1 removes just only the negative supercoils, topoisomerase 2 removes both negative and positive supercoils. Oh, I've never really heard of negative and positive supercoils, so I never knew that, but that's actually pretty cool. Okay. Negative and positive supercoils. I'm not sure what a negative supercoil would be, but if it does that, that's good to know too. So topoisomerase 2 has two functions. Alright. Primase is extremely necessary. You can't really start DNA replication if you didn't have a beginning portion. So primase, this little yellow thing right here, you could think of as, when you're zipping up your jacket, you have that little bar that you have to hook into before you can start pulling it up. That's exactly what primase is doing, right? Primase is making that little bar that you can hook your zipper, which is pretty much your polymerase, right? You can hook your zipper into that little bar, and then you can start pulling it up, right? Without doing that, you can't start pulling up your zipper. So zippers are heavily actually, sort of designed right after the way these guys work, right? So your DNA polymerase 3 cannot start zipping up if you don't have your primase there, right? So primase is making an RNA primer on DNA, right? So remember, this is DNA replication. If you have an RNA primer, that's not something you can just keep there, right? Because an RNA primer on DNA makes it not be completely DNA. So we're going to have to remove that later on, and that is actually taken care of by the DNA polymerase itself, right? DNA polymerase 3, prokaryotes only. Remember, for eukaryotes, you pretty much just have DNA polymerase. That's it. But prokaryotes have DNA polymerase 3, DNA polymerase 1. There's many others as well. DNA polymerase 3 elongates the leading strand by adding more nucleotides to the 3' end and elongates the lagging strand until it reaches a primer for the next lagging strand, right? DNA polymerase 3 has 5' to 3' synthesis, just like all synthesis. There's no other way to synthesize. And it proofreads backwards, 3' to 5' exonuclease, right? So proofreading backwards. Synthesis is always 5' to 3', right? But this DNA polymerase 3 thing is very unique. It proofreads with 3' to 5' exonuclease activity. So it goes back to delete something. Drugs blocking DNA replication often have a modified 3' hydroxyl group, thereby preventing addition of the next nucleotide, leading to a chain termination. Right? The 3' hydroxyl group is extremely important. That's kind of like the attack target for adding primers, right? The 3' hydroxyl group of, let me see if I could actually find a picture of that. So, no, there's no picture for it. A 3' hydroxyl group. No, I don't have a picture for that. So you just gotta remember it though. You do need to remember it. Modifying the 3' hydroxyl group kind of messes up the target that you're gonna attach onto, and without that, you can't continue this chain. All right. DNA polymerase 1 is different from DNA polymerase 3. Its job is to degrade the RNA primer, right? It degrades the RNA primer, it gets rid of this, and it also does everything else DNA polymerase 3 does. But DNA polymerase 3 cannot degrade RNA primers. So DNA polymerase 1 is very special for that. And on top of that, it also has, the way it does that is 5' to 3' exonuclease. You see the difference in here? Proofreading is done backwards, right? But degrading an RNA primer and replacing it with a new DNA area is 5' to 3' exonuclease. Different ways of doing it, okay? So I always remember proofreading is backwards. That's the only thing that happens backwards. Degrading an RNA primer and replacing it with DNA is going to be forward, because that's kind of like doing synthesis. It's the same thing, right? You get rid of the primer and you synthesize new DNA on top of it. Right? And that's kind of what you're seeing over here, right? You see this little fragment, the primer got kicked out, the yellow brick part got kicked out by DNA polymerase 1, and you added a new DNA thing. Right? Now there's a little line here showing you that there's, a lack of connection. That gets taken care of by ligase. Right? Ligase joins Okazaki fragments and links everything together, making sure it's all very stable. The last thing here is telomerase, which is related to telomeres. It's really only found in eukaryotes. It's the only thing on this page that's about eukaryotes only. It's a reverse transcriptase. Right? A lot of people don't remember that telomerase is a reverse transcriptase. It's a reverse transcriptase, which means it's an RNA-dependent DNA polymerase. Right? RNA-dependent DNA polymerase. It's the other way around. Usually you take DNA and make RNA. But here, telomerase can take RNA and make DNA. So telomerase is an RNA-dependent DNA polymerase that adds DNA tags to the 3' end of chromosomes to avoid loss of genetic material with every duplication. And that's actually something that many people don't understand either. But point is, every time you do DNA replication, at the very end, some DNA can get lost. So what telomerase does is it adds extra DNA that you can afford to lose without actually losing valuable DNA. Right? So telomerase tags for greatness and glory is a way to remember the double T, A, triple G. Alright? That's it for that. Alright, what are you guys talking about here? This is the top one. Okay, the linking number of a DNA is the sum of the number of twists of a DNA plus the number of rights. Thus, L equals T plus W. I've never heard of that either. Okay. Okay. Well, I mean, antibiotics don't really work on viruses, right? Because antibiotics are going to target things that viruses don't have. Antibiotics, per se, really only target like cell membrane or organelles or organelle function, and all of those are things DNAs don't have. Viruses don't have. Viruses do not have, per se, a cell membrane or any kind of organelle or any functionality of like, like ribosomes. None of that. So you can't really use antibiotics against a virus. To target viruses, you have to target protein. You have to target amino acids, because viruses, at the end of the day, are just DNA or RNA or some kind of protein. That's it. There's nothing else really in there that's of value. Alright, great. Yeah, people are connecting. Excellent. Studying together is always nice. It's always nice. Just make sure, if you don't feel comfortable, and you want to find someone you are happy studying with. So you keep finding people until you find the person you're perfectly studying with. Alright, mutations in DNA. Now these things are pretty straightforward as well, but again, everything straightforward has a way to become confusing. The first thing they talk about is transition versus transversion, which, way in the beginning it was a little confusing to me. Not exactly confusing, I would forget or whatever, but now it's pretty straightforward. A transition usually means you're doing the same thing still, but in a different way, right? So transition purine to purine or pyrimidine to pyrimidine. That's it. It didn't convert, which is why transversion makes sense. Transversion is a conversion, like, you know, converting religions or something. You were a purine, now you're a pyrimidine, or the other way around. Right? So that's a transversion. That's one technical terminology thing that's down the drain. Then you have silent mutations, missense, nonsense, frame shift, and splice site mutations. The easiest of these are the nonsense mutations. Some nucleotide substitution resulted in an early stop codon. So maybe originally it was like UAC, and then that C became a G, and now it became a stop codon. Right? So normally a wobble position change wouldn't really cause a major effect, but if your first two letters are U and A, it can. Right? Especially, and then UGA. You do need to memorize your stop codons. You have to know these by heart. Right? There's no questioning about it. And people make up all kinds of, you know, statements to memorize it. So whatever you want to use, just go ahead and use it, but make sure you remember them. Right? So if you have a stop codon that suddenly pops up, right? So look at this image that I put right here to kind of see what's happening. Right? In a nonsense mutation, you were going along and then all of a sudden you stopped. So you didn't really finish translation. Well, you didn't finish transcription, so you're not going to finish translation, and you're not going to have a complete protein. In fact, you might even just have a very tiny mRNA that never even goes to translation. The other ones, silent mutations, are super easy. Silent mutation is basically a change in the third position of the codon, right, or the first position of the tRNA wobble position. Right? So this tRNA position wobble, as we saw earlier with the purine, with the example of what was it? This thing right here with the proline example, right? So if you change the U, C, A, or G into anything, all four of them make proline, so there's no issue there. So a silent mutation with proline is pretty common, right? So basically, you had a change in your actual DNA sequence, but you didn't change the amino acid you're making, and that this silent mutation, this is only possible thanks to the fact that your entire thing is degenerate, right? The genetic code is degenerate. So degeneracy is actually kind of coming in pretty clutch, because if it wasn't degenerate, then every single amino acid would only be made by one specific codon, and if you don't have a wobble position thing going on anymore, then any change will lead to a different codon, and that would be kind of bad, right? A different amino acid. Different codons would lead to completely different amino acids, and that would be bad. A missense mutation is where the nucleotide substitution resulted in changing of an amino acid, right? But the new amino acid is very similar to what you had before, right? So examples include sickle cell disease, where you have substitution of glutamic acid with valine. Now these guys are not similar, right? So this is a missense mutation, right? But if it's a conservative missense mutation, then it's because they're similar. Right, so sickle cell is due to a missense mutation, and that you can kind of see right here, right? So this third example, missense, right? DNA has all, and then the sentence no longer makes sense over here because this is not a real word. Can ask for it, so it's not the same anymore, right? The only real way you can make this work, well, it can't work with the sentences I have here, but basically you would need something that sounds pretty much the same. Very much like changing a U with an O, kind of sounds the same. So a missense mutation can be silent, which would just be a conservative missense mutation. But you know, you gotta see what the question is really giving you. Now frame shift mutations are really bad. Frame shift mutations mess up everything, as you can see from the sentence right here. Right, the original sentence is nowhere to be seen anymore. Right, the whole thing. All the codons changed. All the codons changed. It's just because you either deleted or added some multiple of codons that is not divisible by 3. If you add 3 codons or you remove 3 codons, then you're just removing one amino acid. But if you add 1 or 2 codons or you remove 1 or 2 codons, then you shift over the rest of the nucleotides and you create brand new codons. And that's not going to work the same anymore. A relatively newer thing is the splice site mutation, which is just really related to epilepsy. They love testing you on that. but it can be related to cancers and beta thalassemia, dementia, all kinds of stuff. Right? It's a very rare cause for these things, but it can happen. So basically, you have a mutation on the splice site, and then you retain the introns in the mRNA. Remember, introns are intervening. Exons are the ones that are expressed. So introns are intervening. You need to get rid of all your introns, and that's how you get your mature RNA. If you don't do it, then you don't get mature messenger RNA, and you're going to have a chance of getting epilepsy or beta thalassemia or blah, blah, any of these other guys. Because you're going to make a new protein with some kind of different function, and we are not really going to have any idea on what's going to be different in that functionality. Okay. The lac operon is something that is very high yield because it's a very good way to test you on many things. It's pretty straightforward. Actually, no, it's not that straightforward really, but it becomes straightforward, or you have to make it straightforward for yourself. Classic example of a genetic response to an environmental change. Our bodies do this every day. We respond to the things that are happening in our environment by changing our genetic response. So what do we need to know? You need to know this entire table that's down here, right? The questions you're going to get are going to give you a state. You're going to be told what is available for E. coli, and then it's going to ask you what genes are going to be expressed. That's the lac operon, right? And this is a thing for E. coli, right? E. coli does it. So before we get there, we need to see what E. coli likes. So E. coli likes using glucose. It wants to use glucose. It's the easiest thing to use. If there's no glucose, but there is lactose, right? So this is the one we're seeing first, right? Low glucose, but there is lactose available. Then you're going to have lac genes strongly expressed, right? You have your cap and you have your RNA polymerase, and you do not have a repressor protein blocking its way, so the whole thing just works. Now, if you have high glucose and you have no lactose available, then E. coli is still very happy. It's just not going to express lac genes at all, and it's going to go ahead and use glucose. And since it's just using glucose, the repressor protein is right here, and there's no cap, there's no RNA polymerase here. You're not going to make the lac genes because it would be a waste to make the lac genes. You're going to waste energy and resources. Now, if you have low glucose and you don't have any lactose at all, then you're starving, right? But you're going to try to find lactose, so you have your cap, right? But because there really isn't any lactose available, you also have your repressor protein. The repressor protein is kind of an indicator for how much lactose is available, right? You can see every time you see lactose unavailable, the repressor protein is chilling here. And that's because the repressor protein is actually inactivated by lactose, right? See, lactose hooks into there, now your repressor protein disappears. So the presence of a repressor protein on your lac genes is actually indicative of the fact that you have no lactose, right? Now, the cap is a different thing. The cap is related to glucose, right? Glucose inhibits adenylate cyclase, and this is very important in biochemistry in general. It's also kind of related to how insulin and glucagon work. So glucose inhibits adenylate cyclase because adenylate cyclase would take ATP and make cyclic AMP. But if you have glucose, you have ATP, so you don't need to do this, right? If you do not have glucose, this is how I memorize this, right? If you do not have glucose, then you're going to have cyclic AMP because you're not going to have energy. You're not going to have ATP, right? So without glucose, this inhibition no longer occurs. Adenylate cyclase does its job, and you lose energy. ATP becomes cyclic AMP, right? ATP is energy. So that's kind of what I used for myself to memorize this exact mechanism of things that actually is pretty reoccurring. It's going to happen in more biochemistry metabolism as well later on. So the presence of glucose means, right, when you have high glucose in the second and third case, when you have high glucose, there's no cap because you have high glucose, all right? But as soon as there's no more glucose available, you run out of ATP, you make cyclic AMP, which basically makes CAP, your cap, all right? The cap binds something called the capsid, which actually induces transcription. It upregulates the promoter stuff. It leads to more transcription. It helps, excuse me, it helps your RNA polymerase from doing its job. All right. So that's what we're seeing in the first one. And the first one is the perfect scenario for using lac genes, where you have low glucose and high lactose. You express lac genes. All right. Now in this last scenario, this is the one that they like testing you on. You have high glucose and you have lactose. Both things are there. So you're kind of really rich. So now, remember, everyone prefers using glucose first, right? So you're still not going to need your lac genes because you have high glucose. And the presence of glucose means that you're going to have ATP, so there's no cap, right? No cap at all. But you do have lactose, which inactivates the repressor, so the repressor is gone. And that's it. But you see very low expression of the lac genes because you don't need to do it. You're just going to use glucose. All right. Flashcards really depends on your studying method. Right. So if you use flashcards, if you've used flashcards before and it helped you, then gladly go ahead and do so. There's nothing wrong with them. Like, no one's ever going to tell you flashcards are a terrible way to study. It's really dependent on your preference of studying. So if someone says flashcards are a terrible way to study, it's because it's terrible for them, right? And that's okay. Not everyone studies the same way. I would say flashcards are great for like pharmacology, that's for sure. For pharmacology, it's a great way to like memorize medications and what they do and their side effects and stuff. Pharmacology stuff for sure, yeah. But other than that, you know, like I know online when you Google UWorld questions, you can find a lot of flashcards that have questions on it. And those are good drills. You can even do that on your phone if you're traveling or something. You can do it on your phone as you go to sleep. So, you know, I don't have a UWorld, or I would gladly help you out. I don't have a UWorld account anymore. I just use like screenshots. People, like, I found a bunch of UWorld screenshots online, so I just take questions from there. But the questions I put into my book, I got it from like when I did UWorld a long time ago. Yeah, it's expensive, man. Everything, if you're studying medicine and you plan on coming to America to do like USMLE, all of that is very expensive. It's kind of, you know, unrealistic. I completely agree with you. Even USMLE, like step 1 is $1,000, step 2 is $1,000 each for CK and CS. So it's pretty unreal, but it is what it is, and I don't know how they justify it, but there's really no power that we have. So even UWorld, for example, UWorld, pretty much every medical student buys UWorld, yet they still don't lower the price. It's like they have so many sales on it, why keep it so expensive? But that's kind of what happens when you're a business, right? You make money and then you get addicted to that and you justify it and you convince yourself that you're doing the right thing. So it's hard, but you just, at least you can use a credit card. So there's that. There's definitely that. But at the worst case scenario, you could go ahead and just download images and do that. That would be something you would Google and learn, or just email me and I'll tell you more about that. Writing USMLE step 1 in India. You can write step 1 in India. That's definitely something you could do. Alright, so that's it for that. Down here I have a little note from UWorld where, you know, you just got to remember the 2 distinct mechanisms, which is why this lac operon is special. It's one regulated negatively, right? The negative regulation is by the binding of the repressor protein, right? That's the negative guy. It stops more lac gene expression, even if you have no glucose, right? Low glucose, you have your cyclic AMP and you have your cap, but the repressor protein negatively induces lac gene expression. Then you have your positive regulation, which is this one up here, right? You see the plus sign. Right, so cyclic AMP makes cap, another plus sign, causes upregulation, and that's your positive regulation. So it has 2 distinct regulatory mechanisms for monitoring whether or not you express the lac genes. DNA repair is also a pretty common topic. DNA repair, nucleotide excision repair, base excision repair, and mismatch repair. And I believe the new thing is this thing here. This used to be G2 before. Right, now they changed it to S phase, so you gotta make sure you remember that because you don't want to pick G2 if that acid choice is there too. Man, it feels great to be drinking water now that Ramadan's over. Alright, so I wrote over here that first we have nucleotides, which is the G1, then we have bases being added all along, which is throughout the cell cycle, and finally we fix mismatched bases at the end of S phase, which is your synthesis phase, after synthesis finishes. Right, so that this sentence was kind of showing that these three guys are going in order. Right, so nucleotide excision repair, specific endonucleases release the oligonucleotides containing damaged bases, DNA polymerase and ligase fill and reseal the gap, respectively. Those are exactly what those enzymes do anyway, right? Let me put this away for now. So what does it do? What does nucleotide excision repair do? It repairs bulky helix distorting lesions. That's exactly what you have to watch out for, right? What are bulky helix distorting lesions? Is basically wrong nucleotides being attached to each other. That's it. The wrong nucleotides connected, it's going to create, DNA is supposed to be pretty flat on the top and bottom, but if two things are not connecting correctly, you're going to have a little bump. That bump is what it catches and it realizes what to do about it. Defective in xeroderma pigmentosum, where you have the inability to repair DNA pyrimidine dimers, right? You're not supposed to have pyrimidine dimers. Pyrimidines connect with purines and so on and so on. Pyrimidine dimers caused by UV exposure. Right, so squamous cell carcinoma and melanoma are common complications in patients with xeroderma pigmentosum. Those are things that happen to them, and this is an image showing you squamous cell carcinoma on the nose right there. Findings are dry skin, extreme light sensitivity, and skin cancer. Right, ultraviolet specific endonuclease is deficient in xeroderma pigmentosum. This enzyme recognizes and excises distorted thymine dimers. All right. Photolyase enzyme helps in nucleotide excision repair. Yeah, that photolyase is exactly what this is, the UV specific endonuclease. Right, there's two different guys that are in here. Pretty much any S choice you see, right? Remember, not everything. You're always going to get that one question on step 1 on every single block that's going to have like something you've never heard of. But if you use enough logic and if you use enough of your recall of what you do remember, you should be able to pick out the right answer choice. Because even though you've probably never heard of it, most people do actually end up getting it right. Base excision repair is exactly what's in the name, right? These names are pretty well made. Nucleotide excision repair, you remove a nucleotide and put the correct nucleotide there. The two nucleotides have to be matching, otherwise you're going to have a bump, right? That's that bulky helix. Base excision repair is removing an entire base, right? So base specific glycosylase removes the altered base and creates an apurinic or apyrimidinic site. One or more nucleotides are removed by AP endonuclease, which cleaves the 5' end. Lyase cleaves the 3' end. Then you have DNA polymerase, which fills the gap, and ligase seals it. And UWorld does have questions. UWorld has questions on the order of these enzymes, which is why gel please is an excellent way to use it. It is a lot of names, but these enzyme names are pretty repetitive. Endonuclease and lyase were already talked about. Polymerase has been talked about. Ligase has been talked about. Glycosylase is one of the newer ones here. Right, so glycosylase, what it does is just removes entire bases. That's the special guy in base excision repair. He's new. So it removes non-bulky DNA base alterations. Remember, these are not going to be bulky. Non-bulky DNA base alterations, including depurination, alkylation, oxidation, and deamination. Excessive consumption of dietary nitrites can promote the deamination, causing the following changes that we looked at earlier, right? Cytosine, uracil, adenine becoming hypoxanthine, and guanine becoming xanthine. These abnormal bases can cause DNA mutations, leading to carcinogenesis. So you need base excision repair as well. Last thing here is your mismatch repair. And mismatch repair occurs in the S phase, which previously was not the S phase, so you got to remember that. So your newly synthesized strand is recognized. Now this is the first thing you gotta, you need to stop right here and figure out, whoa, how exactly do I recognize my newly synthesized strand and which one was the old original strand, right? When you're doing DNA replication, right, you see how I'm making a new strand here and it's lighter colored for this image. How will any other part of my body, any other cell or protein, recognize that this is the new strand and not that one outside, right? Because that's what they're saying here, right? The first step of mismatch repair is figuring out the newly synthesized strand. So the way you do that is by looking at the epigenetics, right? And Old strands will have acetylation on it, it'll have methylation on it in different parts, it'll have all kinds of things that have been done to it, editing. Whereas the newly synthesized strand is like a new baby. Nothing has changed on it. There's no acetylation, there's no methylation. And so that's how these guys recognize which one is the newly synthesized strand. Once you recognize the newly synthesized strand, the mismatched nucleotides are removed. Right? You have to recognize the newly synthesized strand, because if you don't, then how do you know that if this is correct or if that is correct? Right? Which one am I fixing? Am I fixing the new guy, which is obviously the case? You have to fix the new guy because the new guy is wrong, or mess up and then you end up fixing the old guy and you keep the mistake that the new guy did. So you don't want that, right? Mismatched nucleotides are removed and the gap is filled and resealed. And you see they don't mention the names of all these enzymes again, because you should know it by now, right? The gap is filled by polymerase, it's resealed by lyase, and so on and so on. Removing is going to be done by glycosylase. Occurs predominantly in the S phase of the cell cycle, which is pretty much the entire time you're doing synthesis. This is defective in Lynch syndrome, which is hereditary non-polyposis, colorectal cancer. Yeah, with the mut-S, mut-L, and mut-H group. Those are the guys that it comes up again when we talk about gastrointestinal, when we talk about colorectal cancer over there. We're going to see these mut-S, mut-L, and mut-H group mismatch repair proteins, because that was actually, not only is it in first aid, but that was actually an answer to a question in UWorld as well, right? So mismatch repair is achieved by the 3', 5' exonuclease proofreading activity of DNA polymerase 1 and 3. Remember that the only time exonuclease activity was from 5' to 3' was when DNA polymerase 1 was excising primers, right? It's very important to keep repeating things to yourself without actually looking at the material, so you can master it, like I just did right now, right? I didn't go back to look at, oh yeah, what was the different thing about DNA polymerase 1? I remember it. DNA polymerase 1 excises primers, and it does it by 5' to 3' exonuclease activity. And it's 5' to 3' because excising primers and putting in the correct DNA stuff is kind of like doing synthesis. So that's it. It's so hot today. Alright, excuse that little break. Okay, so where are we? How does the endonuclease recognize GATC sequence? Endonuclease, right? I have no idea. I've never... Endonuclease recognizing the GATC sequence. I mean, that's probably what its function actually is, but I'm not sure how it would recognize that other than actually looking at it. But I haven't heard of that before either. Man, so many things that people don't really know. Like, there's so much I don't know. So, but you know, it's not about knowing everything. It's knowing about the right things and making sure you can figure out the rest. So, if I did see a question about the endonuclease recognizing GATC, I would do my best to figure it out, and hopefully I would. The double-strand fixing part is not exactly the most important. Very low yield. I've actually never seen a question on these things, which, you know, again, could mean that you're going to have it on your test, but I really don't think so. Non-homologous end joining and homologous recombination. Just notice from their names, non-homologous, so there's no need for homology. Anyone can be end joined, right? The ends are being joined. That's all that's happening. So they don't have to be homologous ends. Whereas homologous recombination requires two homologous DNA duplexes A strand from the damaged double-strand DNA is repaired using a complementary strand from the intact homologous double-strand DNA as a template. Restores duplexes accurately without a loss of nucleotides. Right? And this is defective in breast and ovarian cancers with BRCA. Right? BRCA1 specifically, right? And in Fanconi anemia as well. Right? So Fanconi we see more of in renal. So homologous recombination not working, is related to these guys, but I've never really seen a question. Even with all the questions you have about BRCA and breast and ovarian cancer and Fanconi, I've never seen a question about homologous recombination. So there's that. Start and stop codons you just need to know, as I said earlier. Right? These are some words that people use for the stop codons: UGA, UAA, and UAG. So these are 3 different sentences people use for that. The start codon is always AUG, so that's already pretty set. Not much to talk about there. Just remember that stop codons, the way they stop something is these do not really code for amino acids, right? UGA does not make an amino acid. Instead, they're recognized by proteins called release factors, which then terminate translation. The VDJ recombination stuff is going to come up later on in immunology. Right? VDJ recombination also uses non-homologous recombination, so issues with non-homologous end joining will lead to SCID. Right? And ataxia telangiectasia is also something that we're going to see later on in immunology as well. These VDJ recombination is, honestly, I can't really remember too well right now, but it's related to the way we are able to make all kinds of antibodies. Right? We can make all kinds of antibodies from a limited set of resources. It's very similar to how splicing allows you to take a little bit of DNA but make all kinds of mRNA, right? Because based off of where you spliced it and where you reconnected it, you make different kinds of mRNA. So VDJ recombination allows you to change up your antibodies to make all kinds of new antibodies for different diseases. The way this is different, though, is that this uses non-homologous end joining. Give me 1 second. Mom is calling. Hello? हाँ क्या हुआ? हाँ नहीं अभी तो नहीं आ सकता. ठीक. Mom just came home with groceries. I can't help. But my sister's with her, so she'll take care of it. Alright, so yeah. But we're definitely going to see a lot more about the VDJ recombination when we get to immunology later on. Okay, let's see what's what was that? Fanconi anemia. Fanconi anemia is just an anemia that affects the bone marrow. It's inherited. It's an inheritable anemia, and it's also a rare kidney disorder. But you can definitely find it in first aid and look more into that as well, or Google it. But it has a heavy relation with homologous recombination because it does occur from not fixing a double-stranded DNA issue. So that's something that it's related to each other. Let me see what's happening here. That image is supposed to be this. Oh, okay. This was a big image, so I couldn't really fit it anywhere else. But it was related to this. So let's talk about this first. RNA polymerases. We talked about DNA polymerase 1. We talked about DNA polymerase 3. Now we're looking at RNA polymerases. RNA polymerases can be very confusing for many students. A lot of people tend to forget what's happening. These highlighted guys actually come up pretty often as well. So RNA polymerase 1 makes rRNA, which is the most common, right? The ribosomal RNA, and is present only in the nucleolus. Very important little fact right there, right? Present only in the nucleolus. So if you ever get a question on someone finding RNA polymerase 1, well, rRNA are in the densely basophilic nucleolus. The rest are found throughout the nucleus. Alright, so RNA only in the nucleolus. RNA polymerase 2 is the one that's pretty much everything else, right? It makes mRNA, it makes small nuclear RNA, right? And we already know that mRNA is red 5' and 3', right? So that's RNA polymerase 2. RNA polymerase 3 starts with a T, right? And that's for tRNA, okay? The 5S rRNA, not exactly a big deal to really remember. If you get a question on this, hopefully you'll remember it's RNA polymerase 3, but it would be fucked up to ask a question about that anyway. So no, there's no. Our viruses almost never have proofreading activity, which is why they mutate so much. So no proofreading function, but can initiate chains. RNA polymerase II opens DNA at the promoter site, and we saw that as well, right? So RNA polymerase II is pretty important. It's pretty critical. It's related to messenger RNA, and it is also the one that opens DNA at the promoter site, creating your origin of replication, which was dead smack in the middle, right? So right here, point A, right? Point A, origin of replication. Right here. And RNA doesn't really need proofreading, because if you make a bad mRNA, there's no issue. You're just going to make one bad mRNA or two or three, whatever. You're still going to be making a lot of good mRNA as well. So hopefully the one or two bad mRNAs you made will only make a few bad proteins. There's no issue to it, right? So 1, 2, and 3 are numbered in the same order that their products are used in the protein synthesis, and so on and so on. Alpha-amanitin, which is found in Amanita phalloides, the death cap mushroom, inhibits RNA polymerase II. So now, right now, you can no longer really open DNA at the promoter site, and you cannot go ahead and make mRNAs. You can't do protein transcription anymore. And without the transcription, you get a lot of issues everywhere. But generally, because you eat this and it's part of the GI system and all of that, it causes severe hepatotoxicity, right? Severe hepatotoxicity. And of course, your liver is responsible for making many proteins, right? So it's not going to be able to make it anymore. So that's a pretty big deal. You got to watch out for that. Actinomycin D, which also known as dactinomycin, inhibits RNA polymerase in both prokaryotes and eukaryotes. So although you can give this as a medication for the prokaryotic part, it also harms the individual that's taking it with many side effects. There's a mnemonic for it, right? Children act out. So you kind of give this for childhood cancers as well. Cancers that children get. Very specific ones, but I can't really recall them yet, so that's why it's going to be a good thing that we're going to go through immunology. But yeah, actinomycin D is something that you can use for treatment of cancer, because it messes up the formation of RNA, the use of RNA polymerase. So without RNA polymerase II, you can't really go ahead and replicate more DNA or make mRNAs very well. So actinomycin D is pretty clutch there. In prokaryotes, you only have one RNA polymerase, but it's a multi-subunit complex. Right? So one RNA polymerase in prokaryotes, but it was the other way around when it came to eukaryotes. Right? For eukaryotes, we have multiple RNA polymerases, but we have one DNA polymerase. Prokaryotes have multiple DNA polymerases. This one RNA polymerase makes all three kinds of RNA. Rifampin inhibits the DNA-dependent RNA polymerase in prokaryotes, right? Very important medication. So now you know what else it does in terms of mechanism. Let's go back to what we saw up here. So this table was from UWorld, related to the same RNA polymerase as we just saw, right? So RNA polymerase 1 was basically all the ribosomal RNAs you can care about, and they're found in the nucleolus. RNA polymerase 3 is everything else that's pretty important: messenger, small nuclear, and microRNA. Right? And remember, microRNA, they're used for gene silencing. We're going to see more about that as we go on. RNA polymerase 3, the only thing it really makes is the other T guy, which is a transfer RNA, tRNA. All right? Functional organization of a eukaryotic gene is pretty important. This image kind of got a little blurry. You should be able to find it online too, though. It's just there to name a couple vocabulary words, right? So you have your sense strand, your anti-sense strand, your mRNA, and your peptide. So you do need to know these words, because in a question you could be given, they could be like, okay, so what happens to the sense strand? And then if you don't know what the sense strand is or what they're talking about when they mention a sense strand, then you're gonna have some trouble there. So the sense strand is the one that you see on the top, 5' to 3', right? Your anti-sense strand is the second strand that's with that same DNA. So you have your double-stranded DNA. You do transcription of the anti-sense strand. And this makes sense, because why do you do transcription of the anti-sense strand? By doing transcription of the anti-sense strand, you're going to make sense. You're going to make the same exact thing that you have on your sense strand, the only thing that changes is now you went from DNA to RNA, so all your T's became U's. Right? And then you can see AUG, which is your start codon, right? So that's how I memorize this. Transcription is done for the antisense strand, and when you do transcription of the antisense strand, you make sense. All right? When you make sense, that the way I'm saying that is because you're making a strand that looks just like your sense strand, right? And by doing transcription of your antisense strand, you're pretty much expressing the sense strand, right? So now you have your mRNA, then you do translation of it, and you get your peptide. Right? So these peptides are actually a direct result of what was on the sense strand. Okay? So that's one of the vocabulary things over there. Over here, what you're seeing is you got this long strand of DNA going from the 5' untranslated region all the way to 3' untranslated region. Right? You have things in front of it, and they can be very far away in front of it as well. All for the promoter part, right? So you have your cat box and your tata box. And you can see that the ATG or AUG start codon thing is right after the promoter, but still within your 5' untranslated region. Right? It's not actually part of the exon. It's not one of the exons. Right? It's just part of the untranslated region. And you can see right. Well, it is in the exon per se, but it's also part of the untranslated region. Right? It is in the exon, because you do express the AUG and you make methionine, right? It does happen, but it's also got its own thing, right? So transcription starts in the untranslated region. You have your start codon, you have your introns in between, and you can see there's a very specific thing that's happening where the introns start and end, right? Intron starts at GT and ends at AG, so the G is pretty clutch for figuring out where your introns are, right? And then recognizing that GU and the AG is exactly how introns are actually removed in splicing, and we're going to see that soon enough as well. So you had your long strand that became a very tiny protein coding region, right? Only the pink stuff, that's all you needed, right? And this is mature mRNA because it's been processed. You have your 5' cap, right? The guanosine cap, the G cap. You have your poly A tail. And then when translation occurs, you're going to have your tertiary or quaternary structure based off of how big it is. What are translation factors? What do you mean? Alright, so that's... Oh, and notice how the silencer is on the opposite side of the promoter. Right, so silencers are usually way after the 3' untranslated region. This is, actually they're on both sides, but the only thing that you have after the 3' untranslated region is usually going to be a silencer. You're not going to have a promoter here. You're not going to have an enhancer here. In the beginning, you can have anything. You can have an enhancer, a silencer, or a promoter. But after the 3' untranslated region, you're usually only going to have a silencer. There's no enhancer or promoter after the 3' untranslated region, which kind of makes sense, right? So regulation of gene expression, right? Promoter, enhancer, silencer. What's the difference here? A promoter is a site where RNA polymerase II and multiple other transcription factors bind to DNA upstream from gene locus, usually the AT-rich upstream, so the TATA or the CAT boxes. Promoter mutation commonly results in a dramatic decrease in level of gene transcription, which, not the worst thing on earth, but it's pretty bad, right? Let's say there's a protein you really need to make a lot of. If you're not making enough of it, then that's problematic. Enhancers and silencers. Oh, it's getting really hot in here. Let me know if this fan is making too much noise. I really hope it doesn't. Alright, so enhancer and silencer. An enhancer is where a DNA locus regulatory protein activator binds, right? So you increase the expression of a gene on the same chromosome. This is different from a promoter because it does not require the AT-rich upstream. This is not an AT-rich thing, right? It's a part of the DNA that has regulatory proteins which actually actively upregulate expression, right? So the promoters are a little more passive. Because there's a lot of A's and T's, it's really easy to go ahead and do more transcription, translation, and all that. A silencer is very much like an enhancer in the sense that it's also actively going to stop and slow down expression, right? So you make repressor regulatory proteins, right? So enhancers and silencers are like, they're actual people, they're actual proteins. They go out and either enhance or make things work better, or they silence and stop things from working well. A promoter is passive. Just because you see it, it goes ahead and works better, right? And just by being there, it causes more gene transcription to occur, right? So all of these guys are related to gene transcription, which then obviously equates to translation. RNA processing. Now there's two questions here on RNA processing, because this is a pretty important topic that everyone needs to master pretty well. Eukaryotic translation factors required in forming initiation complex. No, you don't need to know those that well. No, just know like the basics, not like hardcore stuff. We'll see that too. We're almost there. So RNA processing in eukaryotes. Initial transcript is called a heterogeneous nuclear RNA. That's the first RNA you made, right? Now that has to be modified, and that's going to become your mRNA, which is your mature RNA, right? It's messenger RNA, but we're also talking about mature RNA. The following processes occur in the nucleus. So you have your capping of the 5' end, which is basically the addition of a 7-methylguanosine cap, polyadenylation at the end of the 3' end, right? So you see your polyadenylation. You have your hydroxyl there as well, and you have to splice out all your intervening introns so that all you have in between is the coding exons, all right? The capped, tailed, and spliced transcript is called mRNA. mRNA is then transported out of the nucleus into the cytosol, where it is translated. mRNA quality control occurs at cytoplasmic processing bodies known as P-bodies, which contain exonucleases, decapping enzymes, and microRNAs. mRNAs may be degraded or stored in P-bodies for future translation. Right, so P-bodies, you can easily get a straight-up question on that as well, where all that's doing is if you made a mature RNA, but you don't need to translate it right now, you can either keep it in the P-body for later when you might need to translate it, or you can just delete it, break it down. That's what a P-body does, right? So it's a processing body, it's also a preserving body, you can call it whatever you want. But its real name is a cytoplasmic processing body, all right? I do not know about holiday junctions, no, but do tell. It's always good to know these things. What the hell is a holiday junction? So one important thing you want to watch out for is that they told you all of this occurs in the nucleus, right? So I wrote over here, mitochondrial genome will never be capped or tailed or spliced, right? Any RNA that came from anywhere else besides your own nucleus will not be mature, in the sense that it's not going to be capped, tailed, and spliced, all right? In an experiment, cultured fibroblasts are mechanically lysed, and the membrane lipids and cellular proteins are chemically removed to isolate nucleic acids. All right, cool. So we're breaking everything down just to find the nucleic acids. Very important to do what I just did, right? You read a sentence, it might look really confusing. Make sure you understood what happened there. Lysed, we just broke down a bunch of fibroblasts just to isolate nucleic acids. All right, perfect. The cellular extract containing the purified nucleic acids is incubated along with short sequences of repeated deoxythymidine residues fixed to latex beads. So we mixed all the nucleic acids we got with a bunch of deoxythymidine. Put it all in the same place. Mix it all up. The solution is washed several times to remove unbound molecules. Which of the following types of nucleic acids is most likely to bind the strongest to the latex beads, which have the thymidines? And the correct answer was mature mRNA, which 28% of people got. The other 28% of people chose promoter regions of DNA. The adenine residues of the poly A tail would form complementary base pairs with the deoxythymidine residues on the beads. Right? We took a bunch of nucleic acids, we mixed it all together with a bunch of T's. So it's like a poly T bead. That's what that bead is, right? It's a poly T bead. A poly T bead is going to bind with the poly A tail. A and T, they bind with each other. So that's why mature mRNA was the correct answer. Binding of these with your DNA does not make sense, right? This poly A chain of adenine residues is not transcribed from DNA, so these thymidine would not bind DNA. DNA doesn't have a poly-A thing going on. There's no poly-A tail on DNA. The poly-A adenylation was something that occurred after you made your RNA, right? It's part of the processing. So DNA promoter regions have TATA and CAT or a GC box, but these would not be significant for a poly-TB. So that's what was happening there. Okay. Alright, so then there's this question right here which says, a 32-year-old man is recovering from extensive burns. Fibroblasts near the site of injury actively synthesize precursor mRNA to be used as templates for protein synthesis. After transcription, extensive processing of the precursor RNA occurs to form finalized mRNA sequence. The finalized mRNA then exits the nucleus and undergoes translation by ribosome complexes before being degraded. Which step of processing and handling of mRNA occurs only within the cytoplasm of cells? And the only thing that really occurs regarding handling or processing of mRNA in the cytoplasm is the P-body. Otherwise, all other processing of RNA occurred in the nucleus, right? The only time you do any processing for RNA in the cytoplasm are your cytoplasmic processing bodies, which are your P-bodies. All right, so that was a pretty easy question, but it's really important to make sure you understand that this is something that's in UWorld, so it's definitely going to be something that you can get more questions on in your actual step 1. We talked about that. Now here we have your splicing. So when we looked at the splicing thing that was happening, we did see over here, right, the GT and AG, which when you got into RNA were becoming GU and AG because there's no T, right? And the GU and AG were there because those guys are actually pretty important in recognizing where the intron you had to remove is, right? So look at why that was important. Now you have your U1 snRNP and your U2 snRNP over here, right? And what do these guys do? They're actually there. U1 is attaching on the 5' splice site, and U2 is attaching on some branching point, right? It takes this and makes something that looks pretty much like a heart, and you have your spliceosome, right? You took this 5' site and you attached it to the branching point. Right, so you have your hydroxyl group now, which is going to do, remember these arrows that you used to make in organic chemistry for how things are going to react with each other? So this 5' hydroxyl is going to attack this phosphate group over here, and you're going to combine the 2 exons and remove the intron. That's it. Right, so your G's were the ones that were pretty critical for being able to remove an intron correctly. Now if you get a question where the G was mutated, right, or you deleted the G somehow, or you added extra G somewhere, etc., that can heavily influence whether or not a spliceosome is going to recognize the intron and remove it. And if you have splice site mutations where you mess up the splicing, then remember that's related to your epilepsy, dementia, and all those guys, right? We talked about earlier, splice site mutations. So now look at this question right here. Spliceosomes remove introns containing GU at the 5' splice site and AG at the 3' splice site. Oh, sorry, wait, that's not the question. This is actually the information I was just telling you guys, right? So the GU at the 5' and the AG at the 3', all of that, everything gets removed, and you finally have mature RNA. Now this is the UWorld question. So, and this image was given with it. A 6-month-old boy is evaluated in the clinic due to delayed motor development. The mother reports that the boy is weaker than other children his age. He can barely lift his head up when prone and is unable to roll to the side. Physical exam shows generalized hypotonia and decreased deep tendon reflexes. Further evaluation reveals mutation of a protein involved in the assembly of small nuclear ribonuclear proteins in motor neurons. Alright, he most likely has impaired function of what cellular elements? Now, all you have to remember is these SNRPs. The SNRPs are exactly what do splicing, right? They're there for splicing. They're the ones that are right here, your U1 SNRP and your U2 SNRP. So without them, you're going to have an issue with spliceosomes. This infant with delayed motor development and flaccid paralysis likely has spinal muscular atrophy. It's caused by mutations in survival motor neuron 1 gene, which encodes a protein involved in assembly of SNRPs in lower motor neurons. Defective SNRP assembly results in impaired spliceosome function and degeneration of the anterior horn. So that was a little lesson right there in the explanation about spinal muscular atrophy. But the way this question was worded, you got to remember, anytime you got a question on SNRPs, anytime you got a question on what is it? Oh, actually, you know what? This is a nice question. In this question, they only gave you one thing. They talked about SNRPs, and they expected you to know about splicing. UWorld tends to do this thing where they mention way too much information and pretty much just give away what's wrong, right? So that's something that NBME doesn't tend to do. NBME will just be like, mention one tiny detail and then be able to expect you to be able to get like an entire diagnosis down, right? So you gotta know what you're looking out for. Now it looks like this image wasn't related to this, but this image is pretty important. An image like this was given, I can't remember what it was for. Oh, I think it was actually related to the freed 3' hydroxyl, blah blah blah, joining exons. In one question, okay, so right here, in one question, a guanine becomes an adenine in this gene sequence where exons are capital letters, right? So the capital letters are all your exons, your lowercase letters are your introns. And you see this highlighted lowercase g, that's going to become an A in that question, right? And that would cause incorrect splicing of pre-mRNA. And this is something, you know, step 1, UWorld, NBME, they all love doing it, right? You get this really intimidating looking DNA sequence, they tell you something went wrong somewhere, and then you start thinking of all the things that could have happened. Is it a start codon, right? The thing that got mutated, is it a start codon? If it's not a start codon, is it a stop codon? Right? Because then you try to figure out all the different mutations you could have possibly gotten. If it's not a stop codon either that got mutated, then did the thing that got mutated, did that become a start codon? Right? If you make a new start codon here, then that can also cause different issues. And then the last thing you always want to think of is the splice site mutation. Did you mess up a G somewhere? Right, because those G's are very critical. GU and AG, they're very important for finding where you're going to do the splicing and where you're going to remove the introns from. This is just showing you, the way you're able to take small amounts of genetic mutation and do something called alternative splicing. Right? Alternative splicing can produce a variety of protein products from a single heteronuclear RNA. You can take the same heteronuclear RNA, which is right here, right? Take the same heteronuclear RNA and splice it in different ways to make different mature RNA. See, over here in this example, they made 3 different kinds of mature RNA, and each one gave you a different kind of protein. Right? So the one same small segment of DNA can give you multiple proteins. And this is why the amount of DNA we have is actually sufficient for making every protein we could possibly ever need. We should probably be able to make even more proteins, but somehow along the road we lost many things and gained new things. Variants in which splicing occurs abnormally are implicated in oncogenesis and many genetic disorders like beta thalassemia, Gaucher disease, Tay-Sachs, Marfan, and even epilepsy, right? So the introns are being, with splice site mutations, there's a lot of new things that are being discovered in medicine. And since that is a pretty up-and-coming topic, you can definitely look forward to that on your step 1. Oh, and also notice I started labeling which ones we're talking about. So SNRPs are RNA polymerase 2. Remember, everything is RNA polymerase 2. RNA polymerase 1 is ribosomal. RNA polymerase 3 is your tRNA. Can you explain about motifs in protein and gene? I don't know why I disconnected for a second, but I'm back. Anyway, the next thing here are microRNAs, right? So these are also RNA polymerase 2. MicroRNAs are a newer thing that are pretty up and coming as well, because a lot of people are starting to target things genetically to start making life better. If you guys haven't heard of Peter Attia, is a doctor here in America. He's a very, very popular guy right now. He talks about CRISPR. He talks about pretty much all kinds of ways to extend life, right? He talks about a lot of stuff. He has really good TED Talks. He has a podcast. He's got a lot of things on. Peter Attia. Peter Attia is amazing. Very amazing guy. When you get a chance, check out Peter Attia on YouTube when you're bored. He will talk about all kinds of things and make amazing sense out of it. No, it is blurry. It is blurry. Give me a second. I don't know what happened, and then all of a sudden, my thing just said it was going to disconnect. That's why I stopped. I'm not talking about the question right now. I'll wait till it says HD now, so let me know if it gets clear. But for some reason, the software I use disconnected for some reason. I'm not sure why, but it should get clear. Refresh your screen if you have to, because I just clicked on the HD button and it did become HD again. I'm watching the same stream you guys are as well, just to make sure everything's good. So the stream does look clear now. Let me know. Okay, cool. Yeah, it gets clear. It just takes time. I don't know why I disconnected. Hi, Tati. Alright, so continuing on, microRNAs are pretty important. What did they do? Now, since not many students really have, complete knowledge of microRNAs, UWorld Step 1 loves picking on this shit. MicroRNAs are small, which, fits the name, non-coding RNA molecules. They are RNA molecules, they're not DNA. You know they're RNA molecules because they're RNA polymerase 2. That post-transcriptionally regulate gene expression by targeting the 3' untranslated region of specific mRNAs for degradation or translational repression. Remember I told you that after the 3' region, there's usually only going to be a silencer. There's never a promoter, never an enhancer after the 3' region, right? And I don't mean never, but generally speaking, there usually isn't. So with this, microRNAs, what they do is they go to the same 3' untranslated region and they start degrading messenger RNAs, right? Abnormal expression of microRNAs contributes to certain malignancies, such as by silencing an mRNA form a tumor suppressor gene. So what we're talking about here is you make messenger RNAs, right? And you made them because you needed it. But if I have microRNAs in me, or if someone injects microRNAs into you, and it breaks down the mRNA you made, then it pretty much silenced your DNA, right? Because your DNA made an mRNA. That mRNA was going to make a protein you needed, but now this microRNA killed that mRNA, so you'll never make that protein. And that's what microRNAs do. So now pharmaceutical companies are trying to make microRNA kind of medications, which actually go into your body and kill mRNA, right? So you never make that bad protein. Let's say there's a bad protein that your body's making somehow, or maybe your body is making a protein that's keeping some bad disease in you alive, right? So that's what this UWorld question kind of hints at. So you see here, a pharmaceutical company is investigating new therapeutic agents for treatment of Burkitt lymphoma, and this is a real deal. This is something that's actually happening. A double-stranded RNA molecule consisting of 21 base pairs is created that is complementary to a region of mRNA encoding C mice. Introduction of this molecule into tumor cells results in a significant reduction in cell growth. Western blot analysis of equivalent numbers of treated and untreated cells is shown below. What process was most likely directly interrupted by the treatment? So they told you they gave this person double-stranded RNA. That's not a real thing, right? It's not a natural thing. So if you give someone double-stranded RNA, what is it going to do? Well, if you have double-stranded RNA and it enters the person's body, that double-stranded RNA is going to separate. But since it's used to being double-stranded, it's going to go ahead and attach to something, and it's going to go ahead and attach to other mRNA, thereby interrupting mRNA translation. That's exactly how microRNAs work. They interrupt mRNA translation, and you can no longer make the protein that mRNA was going to make. Alright, so 23% of people chose DNA transcription, and that couldn't happen, right? No RNA is going to go and stop DNA transcription. That's not how that works. But RNA can anneal with other RNA and destroy it, just like microRNAs do. That's exactly what this question was about. So this is known as RNA interference, and that's an important mechanism by which short 20 to 30 base pair non-coding RNA sequences induce post-transcriptional gene silencing. Types of silencing RNA includes siRNA and miRNA. siRNA are small interfering RNA, that's what that is, and then miRNA is microRNA. The human genome encodes over 1,000 microRNA genes, each one capable of repressing hundreds of target genes. So we actually make microRNAs ourselves as well, and it was more recently discovered that we can do something like that. So after being transcribed, microRNA undergoes processing in the nucleus to form a double-stranded precursor that is then exported into the cytoplasm. There, the precursor is cleaved into a short RNA helix by a ribonuclease protein called a dicer. The individual strands are then separated and incorporated into RNA-induced silencing complexes, also known as RISC. This multi-protein complex uses its associated microRNA as a template to bind complementary sequences found on target mRNAs. An exact match generally results in mRNA degradation, but a partial match also causes translational repression by preventing ribosome and transcription factor binding. All right, so that's what that is. Gotta play video games, man. It's like the only way to get through all kinds of struggles. Alright, so now the last thing here, tRNA. Right? tRNA was an RNA polymerase 3. tRNA has many little nitty-gritty topics about it, but you can get questions on almost any of this, and it's not the hardest thing to know. So the structure of a tRNA is usually going to be 75 to 90 nucleotides, which is not really that big. They're tiny little guys, right? They float around. They have a secondary structure, which is a cloverleaf form. Anticodon end is opposite the 3' amino acyl end, and that's something that you have to know. So if you look at the image over here, what do they say? The anticodon. All right, so the amino acyl is up here, and the anticodon is down here, and that's all they're really telling you. Right? You should be able to completely draw this and label it on your own anytime. Yeah, League of Legends, and this is the Lenovo logo over here. So whenever you get a question on tRNA, you should be able to completely visualize this specific structure at any given day, and be able to answer most of these questions. So if you look at these questions, let's see. Well, I don't have the question yet, but if you look at these things that you can get questions on, you see all tRNAs, both eukaryotic and prokaryotic, have a CCA at the 3' end. Right, CCA 3'. The reason they wrote it like this is because, remember, anytime you have a codon written with no numbers on it, it goes from 5 to 3. Right, so you have your 5' CCA 3'. Along with a high percentage of chemically modified bases. Right, many different chemically modified bases can be over here. The amino acid is covalently bound to the 3' end of the tRNA. Right, so whatever amino acid is going to come is going to attack this 3' hydroxyl group, and that's what we were looking at like almost an hour ago. Right, the 3' hydroxyl group is this. That 3' hydroxyl group is the acceptor for an amino acid. So that's the amino acylation, which is charging. Right, T arm and D arm. You have your two different arms. The T arm contains TWC. I call this a W. It's not a W. But the reason I call it a TWC, and I've mentioned it in other videos, is because here in New York, previously, it changed now, but for the longest time, and ever since I've been in New York, the main cable company here was called Time Warner Cable, TWC. That's how they were known. Now it's called Spectrum, which is different, whatever. But TWC was how I memorized what's on the T arm, right? So it's your ribothiamidine, pseudouridine, and cytidine. This sequence is necessary for tRNA ribosome binding. The T arm tethers tRNA, right? That's all it does. tRNA ribosome binding. So this T arm is just there to bind onto ribosomes. Your D arm contains dihydrouridine residues necessary for tRNA recognition by the correct amino acyl tRNA synthetase. And the D arm is significantly more important because this recognition is what makes sure you attach the correct amino acid for every codon. Right? You don't want to make that mistake of putting the wrong amino acid. So amino acyl tRNA synthetase is the guy that's going to make sure that scrutinization takes place. The acceptor stem is the 5' CCA 3', and that's where the amino acid goes. So we already talked about that. Charging. Charging is done by amino acyl tRNA synthetase by using 1 ATP. Right? And, I don't know how many ATP, but it uses ATP. One unique enzyme per respective amino acid. And binding of charged tRNA to the codon are responsible for the accuracy of amino acid selection. Amino acyl tRNA synthetase matches an amino acid to the tRNA by scrutinizing the amino acid before and after it binds to the tRNA. So before an amino acid attaches here, it'll check the codon down here. Then it'll attach it, and it'll check again, and it'll make sure it's correct. And that's pretty cool, right? That's very important that it does that. Amino acid tRNA synthetase. A mischarged tRNA, so whatever amino acid here is not the correct one for this, a mischarged tRNA reads the usual codon, but inserts the wrong amino acid, right? It should not happen. This should not really happen because it's being checked over and over. However, if your amino acid tRNA synthetase isn't working, or something disabled it, then it can happen. So maybe this amino acid does not match this codon, and your mRNA reads it, so you see AUG on your mRNA. mRNA AUG. This G is the third spot on your mRNA, which correlates to the first spot on your tRNA, and that C is your wobble position. Just wanted to point that out again, right? See the wobble position? But anyway, AUG is supposed to give you methionine, right? Now imagine instead of methionine, whatever's over here is not methionine. Your tRNA is still going to go ahead and put whatever the fuck is over here, because your amino acid tRNA synthetase didn't do its job. And that's what this question is about right here. So the first question says, a series of experiments is being conducted to determine the structure and function of different types of bacterial RNA. Cultures of Staph aureus are exposed to chemicals that lyse the bacterial cells, and the RNA molecules are then extracted. A specific RNA consisting of 90 nucleotides is purified for further analysis. It is found to contain high amounts of chemically modified bases like dihydrouridine, pseudouridine, and ribothiamidine, and its secondary structure arises from base pairing within the chain itself. Right? And that makes sense, right? Because there's base pairing within the chain right here, within the chain right here. It kind of just connects with itself. That's what they're talking about. So you already know they're talking about tRNA. What is the most likely composition of the 3' end of this molecule? And that's all you had to remember. CCA. That's it. The other answer choices that were there were ACC, by the way. Right? And yes, a lot of people picked it. You have to remember it's CCA 3', 5' 3', 5' CCA can carry amino acids. Alright, so that's all that question was about. This one over here is about the tRNA synthetase. So it says, in an experiment, erythrocyte precursor cells are incubated in a medium containing radiolabeled cysteine. These radiolabeled cysteine residues are attached to their appropriate tRNAs by the enzyme amino acid tRNA synthetase. The bound cysteine residues are then chemically modified to form alanine. Alright, so they replace cysteine with alanine. The end product of this reaction is a tRNA molecule that contains cysteine anticodon, but is mischarged with alanine. Right, so your anticodon is supposed to be cysteine, which it is, and the amino acid is supposed to be cysteine, but they changed it and they made it alanine. That's what they did in this question. So now they're like, what is the most likely to occur to this alanine residue during polypeptide synthesis of the alpha hemoglobin? Well, by this point, your amino acid tRNA synthetase had already done its job. It's checked. Then you modified it after that. Now amino acid RNA synthetase is not checking again. It's already done checking. So it will incorporate the wrong thing into the chain, right? So it's it will be incorporated into the polypeptide chain. Any site requiring cysteine will have alanine instead, and that's what's happening in that question. All right. So a mischarged tRNA reads the usual codon, but inserts the wrong amino acid. If you get a question where that's happening, that's because there's an issue with amino acid tRNA synthetase. All right. Protein synthesis. So this is your entire protein synthesis little chat right here. You got your ribosome doing its job of translation. So initiation, elongation, termination. Now initiation, and you have to watch over here for where things are requiring energy, because that's something you get easy questions on. Initiation, eukaryotic initiation factors identify the 5' cap, which was done in the nucleus when you made the mature mRNA. Those initiation factors help assemble the 40S ribosomal subunit, which is RNA polymerase type 1. And then the initiation factor is released when the mRNA and the ribosomal 60S subunit assemble with the complex. So once everything assembles, your initiation factors are gone. And by this point, you've used one GTP, or I don't know how many, but I keep saying one, sorry. It requires GTP, so, whatever it is requiring GTP, though. Just make sure you remember that, because they actually make some good effort in telling you that, right? GTP, gripping and going places. Translocation, translation, blah, blah, blah. ATP was required for tRNA activation, when you were doing the charging thing earlier. Elongation is amino acyl tRNA binding to the A site, except for the initiator methionine. The initiator methionine, the first methionine guy, comes into the P site. Right? That's your start codon, goes in the P site. Everything else comes into the A site. That's also something you have to remember. It's a technical little topic that you can get an easy question on. Then you have rRNA, which are also known as ribozyme, catalyze the peptide bond formation, transfers growing polypeptide to amino acid in the A site, right? You keep adding more and more and more as this thing keeps sliding through. Ribosome advances 3 nucleotides towards the 3' end of the mRNA, moving peptidyl tRNA to P site, right? Everything is going to keep going down this way, and it's all going to keep going into the E site, which holds the empty tRNA as it exits, right? So this growing chain, though, just keeps falling outwards, right? That's how you're releasing the chain. The growing chain doesn't necessarily go in the E site. The only thing that goes in the E site is your empty tRNA, the tRNA that is no longer charged anymore. Termination occurs when you reach a stop codon, right? And those terminating factors are going to recognize it. So the releasing factors, right? Eukaryotic release factors recognize the stop codon and halt translation. Completed polypeptide is released from the ribosome, and that also requires GTP. Right? So everything, and this step over here too, all 3 of these things require GTP. So there's a lot of GTP being used when you do translation, and a lot of ATP being used when you do translation as well, but this is the different part of translation, is when you do the charging of tRNA. All of that is related to translation. In the end, after translation, there's still things you can do, just like after you're making a heterogeneous mRNA. Well, hnRNA. So post-translational modifications include trimming or covalent alterations. The trimming part is not that big of a deal. You either remove the N or C terminal propeptides from the zymogen and you make a mature protein, such as trypsinogen becoming trypsin. So if you ever get a GI question on how trypsinogen becomes trypsin, just remember you're removing the N or C terminal propeptides from that zymogen. The zymogen is basically the inactive form. Covalent alterations include all of these guys. And down here is an image from USMLE RX videos telling you what each of these guys usually does. Right? Now, phosphorylation, I like to think of phosphorylation as an inactivation. Phosphorylation almost always inactivates whatever is being done. Right? And there's multiple ways to think of inactivation. Right? Let's say there's a channel. Let's say there's like a channel and you phosphorylate it and now it opens up and things are coming through. You could look at that as an inactivation as well, where that channel's original job was to make sure things don't come in, right? And then you inactivated it. Of course, it's easier to think of it as attaching a phosphate, activated it, now the channel opened up, things come in. You could think of it like that, but for my own sanity, I guess, I thought of phosphorylation as always inactivating things. You just turn it off. A phosphate is a negative group, it turns things off. Glycosylation increases binding affinity, right? Glycosylation occurs a lot, right? That's what happens to people who have diabetic people. They have a high amount of hemoglobin A1C, and they have non-enzymatic glycosylation everywhere, and that causes problems, right? Hydroxylation is basically when you have hydrogen bonds in collagen. That's really the only place it occurs. It's not a very common thing. Methylation and acetylation we already talked about. Ubiquitination is a way to tag something to be degraded. That's what ubiquitination is about. When you do ubiquitination, you basically tell something to go be destroyed. That's pretty straightforward. The last thing on this page is the chaperone protein. And the chaperone protein is basically, now this is pronounced chaperone protein, by the way. I don't know why I said chat. Chaperone protein is an intracellular protein involved in facilitating and or maintaining protein folding. For example, in yeast, heat shock proteins are expressed at high temperatures to prevent protein denaturing and misfolding. Right? And we use these heat shock proteins when we try to do lab techniques. Right? We're going to see that with polymerase chain reaction. All right, so that's the end of molecular right there. Now we're jumping into cellular. Let's see. Cellular. All right, so cellular cell cycle phases. There's a lot of stuff here too, but pretty. Oh shit! It is actually pretty hot. My bad, guys. I really hope this isn't making that much noise. I'm trying to make sure it doesn't face the laptop entirely, but it's really hot today. What is it today? No, it's 80 degrees. Okay. So there are checkpoints in this entire thing of mitosis and all of that to make sure things are under control. You don't want to replicate too much. You don't want to replicate too little. And you definitely don't want to replicate out of control, because that's exactly what cancer is about. So you have your cyclin-dependent kinases. Now, when we get to metabolism, we'll see what a kinase is, but for the most part, we can talk about it right now. A kinase generally always does the same thing, and what it does is it adds a phosphate. That's what kinases do. The job of a kinase is to add a phosphate and usually turn things off, right? That's what phosphorylation kind of usually means. So cyclin-dependent kinases. So this kinase only works when there's a cyclin. A cyclin is a regulatory protein that controls the cell cycle event, and it is very phase-specific, which means it doesn't work everywhere. It works in specific phases, and what it does is it activates the cyclin-dependent kinases, hence why they're called cyclin-dependent. These kinases don't work if there's no cyclin. When you have the two guys together, you get your cyclin-CDK complex. What they do is they phosphorylate other proteins to coordinate cell cycle progression. They have to be activated and inactivated at appropriate times for cell cycle to progress, which makes sense. Now, you have your tumor suppressor genes, right? Tumor suppressor genes and oncogenes are two different things. Oncogenes, when you turn them on, all you need is one oncogene and you have cancer. A tumor suppressor gene, you usually have two, and these guys are making proteins to stop tumors from forming. With tumor suppressor genes, you have something known as the two-hit rule, where basically both tumor suppressor gene copies have to stop working for you to finally no longer have tumor suppression, and then you can get cancer. Now, stuff can get confusing here. So what's happening here? P53 tells P21 to do something. What does it tell P21 to do? Well, it tells P21 to go stop CDKs. All right, so P53 tells P21 to stop CDKs. CDKs, if they're not working, you will no longer phosphorylate retinoblastoma. If you do not phosphorylate it, you activate it, right? Because phosphorylation deactivates things. So now you have hypophosphorylated or active retinoblastoma. Retinoblastoma goes and inhibits G1 to S progression. Mutations in the tumor suppressor genes can result in unrestrained cell division, like in Li-Fraumeni syndrome. Growth factors like insulin, PDGF, EPO, and EGF bind tyrosine kinase receptors to transition the cell from G1 to S phase. Right? So P53 stops the transition from G1 to S phase, whereas growth factors upregulate the transition from G1 to S phase. And that's a very big distinction that you have to remember. That's kind of how these guys work. Right? And you get questions on tyrosine kinase all the time. What does tyrosine kinase do? How does it work? What does it work with? Right? It works for all these growth factors, and insulin is a pretty big growth factor in a sense as well, because it's anabolic, it makes you big, it makes you overweight, etc. So all of these growth factors and insulin use tyrosine kinase, which has to be a pretty intracellular thing, because this is, happening inside the cell, all this DNA replication and stuff. So that's about it for that. Cell types. Now, RER and SER is really just a read yourself kind of thing. Nothing's important there. Cell trafficking is extremely low yield. I really have almost never seen a question on cell trafficking. Eye cell disease, I've never seen a question on either. I'm not even sure if UWorld had a question on eye cell disease, but eye cell disease is pretty simple. So I have notes here to help you kind of memorize all the things, right? Like, for example, Golgi is the distribution center of proteins and lipids from the ER to the vesicles and plasma membrane. It modifies the N-oligosaccharides on asparagine, and it adds O-oligosaccharides on serine and threonine. So I made up the no-as-though statement where N-O on asparagine and O oligosaccharides on serine and threonine. Right, so that's what the S and T over here are supposed to be for. It's not exactly color coded, but you can figure it out. So that's what this is the thing I made up to make sure I remember this, just in case I get that one technical stupid question. I can make sure I get it correct. And it also adds mannose 6-phosphate to proteins for trafficking to lysosomes. And usually the only reason you send something to lysosomes is for it to go die. Right? Lysosomes are going to destroy it. In eye cell disease, which is inclusion cell disease, and it's also known as mucolipidosis type 2, this is an inherited lysosomal storage disorder with a defect in N-acetylglucosamine 1-phosphotransferase. You just need to remember that name. You don't have to be able to pronounce it. Failure of the Golgi to phosphorylate mannose residues. Right? You can no longer make mannose 6-phosphates, and without mannose 6-phosphate, you're no longer going to be able to tell things to go to the lysosome. So now proteins are secreted extracellularly rather than delivered to lysosomes. And that results in coarse facial features, gingival hyperplasia, clouded corneas, restricted joint movements, a claw hand deformity, even kyphoscoliosis. And this is pretty much going to be fatal in childhood. Right? Eye cell disease is also going to be very similar looking to the Hurler syndrome, Hunter and Hurler syndrome, because they also have a form of gargoylism. Right? This coarse facial features is sort of like gargoylism as well, especially with the gingival hyperplasia. So you really want to make sure you don't confuse the diseases. Make sure you're looking for where the pathology is. What is the pathology? What's not working correctly? Signal recognition particles. So now, these guys, the signal recognition particles and your vesicular trafficking proteins, you can get all kinds of questions on them, but I've just never really seen questions on them. Also, in first aid, it only tells you eye cell disease is inherited, but they don't tell you that it is also recessive. So, really important to remember that. Because just telling you that it's inherited doesn't exactly tell you how it's inherited. Alright, so signal recognition particles. Abundant cytosolic ribonuclear protein that traffics polypeptide ribosome complex from the cytosol to the RER. Absent or dysfunctional signal recognition particles results in accumulation of protein in the cytosol because it won't know where to go. There's no signal recognition. Alright? Vesicular trafficking proteins. COP1, COP2, and clathrin. Now, I have a thing I made up for them, and this image kind of helps you. Right? So, COP1 is from Golgi to Golgi retrograde, right? Or from cis Golgi to the ER. Cis being on the same side as your ER, right? Trans means on the opposite side. So this is all the orange ones you see here. You see it goes retrograde, and then from the same side, cis Golgi back to the ER. So you're bringing something back to the RER. COP2 goes from the ER to the cis Golgi and pterograde, and that's it. Nowhere else, right? There's only two of them here. That's how that's another thing I always thought of. In this image, you only see two COP2s, and all it does is it takes from the RER to the Golgi. That's it. Clathrin takes it from the trans Golgi, which is further away from the RER. Right? It takes it from the trans Golgi and sends it out to the plasma membrane, or it could send it to lysosomes, endosomes, pretty much anywhere. Right, so I made up a thing for, and they have it too, right? COP2 is two steps forward, and 1 is 1 step back. So you can just think of it like that if you want to as well. It definitely works, it helps, and that's generally all that's happening there. For the RER and SCR, you really just want to remember what their function is and have a good idea of how they're different, right? Rough endoplasmic reticulum is rough because there's ribosomes on it. SCR has no ribosomes on it. The smooth endoplasmic reticulum is only there for steroid synthesis and detoxification of drugs and poisons. Right? There's no ribosomes on it. Liver and hepatocytes and steroid hormone producing cells of the adrenal cortex have a lot of SCR. Right? Because all of those cells have steroid synthesis, and the liver has detoxification. Mucus secreting goblet cells of the small intestine and antibody secreting white blood cells have a lot of RER, because RER guys are really there to send things out of the cell and attack or make proteins, etc., etc. And so very important to understand where you're going to see the guys that are being mentioned. Peroxisome, they've added a lot of things over the last 2 years into here, especially with the pathologies they mentioned. Previously, they usually just used to like say the name of the disease and not really talk about it much. So what does a peroxisome do? It's a membrane-enclosed organelle involved in beta oxidation of very long-chain fatty acids and alpha oxidation. Now, the alpha oxidation thing is very specific to peroxisomes, right? Strictly peroxisomal process. If they ever talk about alpha oxidation anywhere, you know they're talking about peroxisomes. Catabolism of branched-chain fatty acid, amino acids, and ethanol. Synthesis of cholesterol, bile acids, and plasmalogens. Important membrane phospholipid, especially in the white matter of the brain. So this part is also important because, of course, if you're lacking peroxisomes, you're going to have some kind of issue in the brain as well. And we can see that in these diseases. Right? Zellweger syndrome is an autosomal recessive disorder of peroxisome biogenesis due to mutated PEX genes. You don't need to know what the PEX genes are. However, because of this peroxisomal biogenesis issue, you get hypotonia, seizures, hepatomegaly, and early death. That's it. What you're looking for is things that are not mentioned, right? The only way to tell Zellweger different from Refsum is because in Refsum disease, the issue is lack of alpha oxidation. Right? So if they don't mention alpha oxidation and it looks like it's a question about peroxisomes, then you know they're talking about Zellweger syndrome. In Refsum disease, without the alpha oxidation, your phytanic acid cannot be metabolized to pristanic acid. So you have a lack of pristanic acid and you have extra phytanic acid. Scaly skin, ataxia, cataracts, night blindness, shortness of the fourth toe, and epiphyseal dysplasia, right? Very weird, all the things that are happening in this disease. And treatment is usually just a diet restriction. Stop eating things that make phytanic acid. So, yeah, that's there. I've got a couple extra notes over here just to let you guys know. Right, to avoid phytanic acid in the diet, you restrict tuna, cod, haddock fish. Basically, don't eat seafood at all. And also grass-feeding animals and their milk. So no cow, no goat, etc. The last thing is adrenoleukodystrophy. Now, adrenoleukodystrophy is pretty well named. The name tells you what's happening. This one is X-linked recessive. So that's pretty unique already, right? The genetic association. X-linked recessive disorder of beta oxidation due to the mutation in ABCD1 gene. Very long-chain fatty acid builds up in the adrenal glands, in the white matter of the brain, and the testes. Then you get progressive disease that can lead to adrenal gland crisis, coma, and death. Right, so pretty bad stuff happening here. You got to make sure things are under control. Without that control, you just have a lot of very long-chain fatty acids everywhere, and that's it. So you see alpha oxidation issue, a beta oxidation issue, and a peroxisome biogenesis issue. That's it. Right. These questions are pretty much almost always going to be very straightforward. Like, you'll know it's a question about peroxisomes. What they're going to try to confuse you with is by having these other answer choices there as well. Right. So alpha oxidation is Refsum, beta oxidation is adrenoleukodystrophy, and Zellweger syndrome is neither of those. If they don't mention alpha oxidation, they don't mention beta oxidation, there's no buildup of long-chain fatty acids, the question is just going to be about Zellweger syndrome. Alright? Proteasomes, cytoskeletal elements, microtubules, not really too much to talk about with these guys. The proteasome we're going to see again later on where we do talk about Parkinson's disease all the way in neurology. It's a barrel-shaped protein complex that degrades damaged or ubiquitin-tagged proteins. Remember, we just talked about ubiquitination earlier. Anything that has a ubiquitin tagged on it will go to a proteasome and get degraded. It is barrel-shaped. I used to have a picture of where is it? No, I don't have it anymore. I used to have a little picture of a proteasome from USMLE RX. I'm not sure why I took it off. Let me see. Proteasome structure. Like, it's literally actually pretty barrel shaped, and it does kind of like open and close. Yeah, like you can kind of see that here. So if you look at this image right here, right, you see how it's got like a lid on both sides, kind of, sort of, right? So these are all different images showing you what a proteasome sort of looks like. Right? They're doing a pretty decent job trying to recreate the imagery. It's just a barrel-shaped organelle. That's literally all it is. And its single job... No, I'm not playing games. Stop it. Why is it giving me this notification? Alright. So the single job of a proteasome is just to take ubiquitin-tagged proteins and get rid of them. Oh wow. So you know when I was just saying I'm not playing games, it didn't show up on the recording, but there was a pop-up on the screen telling me activate video game mode. And I was like, what the heck? Why is that showing up? Anyway, so anytime the proteasome is not working, that is actually heavily implicated in some cases of Parkinson's disease. In Parkinson's disease, there's a bunch of other issues, and there's a very straightforward pathophysiology of why it happens too. But somehow proteasomes are also implicated in Parkinson's disease, so proteasomes can pop up on your test because Parkinson is a pretty high yield topic. But there's a lot more things to ask about Parkinson than ask about proteasomes. For cytoskeletal elements, do not forget the vimentin, desmin, cytokeratin. These guys over here. These guys are very specific to specific things on the question. You can get a question about where would you find GFAP, for example, right? Gliofibrillary acidic protein actually comes up a lot of times when you have questions about neurological tumors. Right? This is actually a pretty good way to identify neurological tumors. So you got to make sure you remember where these guys are used to possibly diagnose something. Microtubules are the ones that are for movement, whereas intermediate filaments are not really for movement at all. And microfilaments, they're for contraction. Right? So cytokinesis is here too, but really when you talk about movement, it's microtubules that are doing it. And that's why you have your microtubule over here. You see a bunch of medications that target microtubules. And all these guys are, you can see what they're for. And by targeting microtubules, they basically kill cells, right? Because microtubules are going to be necessary for not just movement, but also cell division. So if you don't have cell division anymore, you're going to die. The cylindrical outer structure composed of a helical array of polymerized heterodimers of alpha and beta tubulin. Each dimer has 2 GTP. That's easy to remember too, because remember with G for going places and getting places, GTP is also for movement, so that's related to going places. What you don't want to forget is that dynein is retrograde and kinesin is anterograde. So how do you remember that? So for me, it was just remembering that for potassium is K plus, so it's plus. It goes to the it's anterograde. It goes from the negative to the plus side. Dynein must be the other way around. They tell you that keen to go outside and dying to come back home, which also works, right? At this age, it's all about that. I'm always dying to come back home. I don't know about you guys, but like as much as I love going out, I am always eager to come back home and just relax. There's also this association of what the negative and positive ends are, right? So the negative end is near the nucleus, whereas the positive end points to the periphery. And I think these are great ways of memorizing it. It works perfectly fine for me. So I just used that. So anytime you get a question where they're telling you something is being taken towards the nucleus, you know it's being taken towards the negative end, so you must be using a dynein. That's it, okay? You get silly simple questions like that. And this is another image just kind of showing you the molecular movement of things. Negative end, so the nucleus is going to be near here. Positive end is going towards the periphery like the cell membrane. Down here you have a question about ubiquitination and the mention of Parkinson's, right? So this is a pretty nice question, not just for the question itself, but also for the association of Parkinson's. As I told you, it's a pretty big deal. UWorld asked a question about ubiquitination and mentioned Parkinson's. However, I feel like if I'm going to ask a question about Parkinson's, I would ask it on so many other things, not about the ubiquitination aspect of it. 42-year-old woman comes to the neurologist for enrollment in a research study. She has 15-year history of resting tremor, bradykinesia, and cogwheel rigidity consistent with Parkinson's disease. One of her siblings recently started having similar symptoms. Genetic analysis is performed on the patient and her affected sibling. The results show a loss of function mutation in a gene that leads to an accumulation of misfolded proteins. So right away you know misfolded proteins, where are they supposed to go? They're supposed to be tagged with mannose-6-phosphate, and they go to and they get ubiquitinated, right? So this is either going to be a question about ISO disease or something about proteasomes not working, etc., etc. Then they ask you what biochemical process is most likely defective in her, and the only answer you could possibly pick there was ubiquitination. Ubiquitin is a protein found in all eukaryotic cells that undergoes ATP-dependent attachment to other proteins, labeling them for degradation. The proteasome then recognizes these ubiquitinated proteins and uses ATP energy to drive them through its tubular structure, degrading them into small peptides. Ubiquitination plays an important role in many cell functions, including antigen processing, muscle wasting, cell cycle regulation, DNA repair, blah blah blah. Impairment of ubiquitin-proteasome system can contribute to the development of neurodegenerative disorders like Parkinson's and Alzheimer's. Failure of the system to properly degrade abnormal proteins causes protein misfolding, aggregation, and eventual obstruction of intracellular molecular traffic, leading to cell death. Together, the Parkin, PINK1, and DJ1 genes code for a protein complex that promotes the degradation of misfolded proteins via the ubiquitin-proteasome system. Mutations in these genes are each associated with autosomal recessive forms of Parkinson's disease. This is some kind of silly information over here that you can get on an NBME. So, now that you've seen it, you don't need to sit there and memorize it, but at least you've seen it, you've heard it. You mentioned it, I mentioned it over here. So at least now, if you do see it ever again, you'll know where it came from. This is now talking about cilia structure. So remember, cilia was for movement. Right? Cilia, flagella, mitotic spindles for the cell division, axonal trafficking, and centrioles are all for movement and cell division. These are microtubules. So what is up with cilia? So you can see this picture over here. Right? You have your flagellum and you have your cilia. Flagellum helps for like propagating. It's like a propeller. Whereas cilia is more for back and forth, so it helps move things forward or just along like a digestive tract, for example. So what is the cilia structure? You have 9 doublets and 2 singlet arrangement of a microtubule, and that's what you see over here. You have your 9 doublets and 2 singles in the middle. The basal body, the base of the cilium, below the cell membrane, so under here, consists of 9 microtubule triplets. Right, so the structure changes at the basal area, under the cell membrane. Axonimodynine ATPase that links peripheral 9 doublets and causes bending of cilium by differential sliding of doublets. Gap junctions enable coordinated ciliary movement. So this is all happening thanks to dynein arms, right? And if you come back here, dynein was for retrograde movement, going backwards, right? So let's see now. Where does shit go wrong? Well, if you have Carter-Jenner syndrome, which is primary ciliary dyskinesia, then you have immotile cilia due to a dynein arm defect. This is autosomal recessive. Carter-Jenner is autosomal recessive, and it results in decreased male and female fertility due to immotile sperm and dysfunctional fallopian tube cilia, respectively. Right? If the fallopian tube doesn't have cilia, it's not going to be able to move the ovum forward, and if the sperm doesn't have the right cilia, it won't be able to move forward either. It's not going to reach the location it's trying to get to. So now you have an increased risk of ectopic pregnancy because a male whose sperm works fine might reach the egg, but it'll reach the egg somewhere that it's not supposed to find it. Right? The egg isn't moving forward, but the sperm is moving forward, so it's going to go all the way in. You can get a pregnancy almost anywhere now. And remember that the ovum doesn't exactly get released. Let me see if I can show you guys real quick. We're on page 49. Okay. Yeah, right here. So if you look over here, right, the ovulation doesn't exactly send the oocyte into the fallopian tube. It just stays back out here. Right, in this outer area, and that's really bad because since the ovum is released in the peritoneal cavity, if you don't have any cilia in your fallopian tube, it could just stay here rather than getting sucked up, and the sperm is going to make it all the way here because there's nothing stopping sperm from reaching there, and you can have a peritoneal pregnancy, which would pretty much just lead to exsanguination. She's going to bleed to death, so that's pretty bad. Carter-Jenner is pretty unfortunate. Can cause bronchiectasis, recurrent sinusitis, chronic ear infections, conductive hearing loss, and situs inversus, which is pretty pathognomonic. Right, so you can see the heart is usually facing towards the left, but now it's facing the other way. Right? One very critical thing is the low nasal nitric oxide. This is used as a screening test. Low nasal nitric oxide. Cartagena restaurants take out only. There's no dine-in because they don't have dining arms. They don't have any dining arms for the retrograde movement. Now, Cartagena, in terms of presentation, can look a lot like cystic fibrosis, right? So this is what I'm talking about right here. Primary ciliary dyskinesia presents with chronic sinopulmonary infections, nasal polyps, bronchiectasis, and digital clubbing, just like cystic fibrosis's respiratory tract manifestations. However, extrapulmonary features are different. PCD has situs inversus. It has infertility due to immotile spermatozoa, whereas in cystic fibrosis, the infertility is due to absent vas deferens, right? The sperm moves fine. There just isn't any tunnel for it to move. There's no vas deferens there. And normal growth, whereas in cystic fibrosis, you do not have normal growth. There's a failure to thrive. Diagnosis of PCD can be made with low nasal nitric oxide levels and bronchoscopy or electron microscopy of the cilia. Cystic fibrosis diagnosis is made by elevated sweat chloride levels and abnormal nasal transepithelial potential difference, a more negative transepithelial potential difference. Genetic testing is able to diagnose both things, right? Was it a more negative or more positive? I forgot. I should not be forgetting that. We gotta find that now. Cystic fibrosis. Oh my god, what are they mentioning? Oh, right here. Cystic fibrosis has more negative transepithelial potential difference, right? So more negative transepithelial potential difference is your cystic fibrosis issue. Alright, so now, where were we? Okay, cilia structure. So that's about it for cilia structure, nothing else over there. Your sodium-potassium pump, I love this, the pumpkin thing, right? Pump K in helps me perfectly remember that potassium comes in and sodium goes out. And then since pump has 2 P's in it, that reminds me that it's pump 2 potassiums in and 3 potassiums out. This is something that a lot of students have had trouble memorizing perfectly. They would keep confusing whether it was 2 potassium coming in or 3 potassiums coming in, etc., etc. This medication, Ubiquitin, or Ubiquitin, however you want to pronounce it, Ubiquitin is a cardiac glycoside that inhibits by binding to the potassium site. Right, Ubiquitin inhibits the ATPase, the sodium-potassium pump, not the ATPase. Sorry. Well, it is a ATPase. Yeah, it inhibits the sodium-potassium ATPase by binding to the potassium site. Very similar to kind of sort of how digoxin does. Cardiac glycosides like digoxin and digitoxin directly inhibit the sodium-potassium ATPase, which leads to indirect inhibition of the sodium-calcium exchange, leading to increased calcium, which increases contractility. Right, and this also binds to the potassium site outside. That's exactly where digoxin and digitoxin bind. So everything kind of just targets the potassium on the extracellular side. That's how Ubiquitin and digoxin and digitoxin, they all work. So that's really all there is to talk about sodium potassium pump. I guess the only other thing was the way the phosphate works. So the phosphate is on the inside end too. So when potassium comes in, phosphate also goes in. You can see as potassium's coming in, the phosphate is going back into the cytosol area, right? But when the phosphate is on this, sodium is going out. So that was how I kind of memorized whether phosphorylation. What did phosphorylation do? So phosphorylation allows sodium to go out. Removal or dephosphorylation allows potassium to come in. So pump K in. You just, not only do you pump the potassium in, but you also send the phosphate back in as well. Alright, so dephosphorylation. Collagen, you get questions on these all the time. There's a couple high yield, and other than that, you can get questions on pretty much anything over here. So, collagen is the most abundant protein in the human body. The most abundant protein in the world, though, is keratin, right? Because insects have a lot of keratin. Extensively modified by post-translational modification, organizes and strengthens the cellular matrix. So now type 1, 2, 3, and 4. Type 1 is the most common. It's in bone, skin, tendon, dentin, fascia, cornea, and late wound repair. So this late wound repair is pretty much anywhere, even in MIs. Remember myocardial infarction, the progression of an MI, how it repairs. In the beginning, you have granulation tissue, which is type 3. Later on, after the 2-week period and stuff, within months, you have the late wound repair, which is your type 1 collagen. Alright, type 2 is cartilage. That's really all it is. But for the eye, you got to remember, type 1 had your cornea, whereas type 2 is the vitreous body. Right, so the cartilage, think of it as really soft stuff. So is the vitreous body, is very soft. And the nucleus pulposus is also pretty soft. Alright, nucleus pulposus gets tested pretty often, right? So you got to remember that it's a type 2 collagen. Type 3, now 3 is skin, blood vessels, uterus, fetal tissue, and granulation tissue. So uterus and fetal tissue, type 3 collagen. Right? Skin is usually just type 1. You don't really want to think of skin for type 3. Skin is usually just type 1. Blood vessels are type 3. And uterus and fetal tissue are type 2. Granulation tissue and late wound repair, you got to remember from MIs. We're going to talk about it a lot more in cardiovascular as well. And then 4 under the floor just reminds you of basal membrane. Right? It's targeted in Goodpasture syndrome and is defective in Alport syndrome. Right? So it's a big deal there. Now collagen synthesis and structure, this, like, you almost are guaranteed to have a question on this on your step 1. So steps 1 to 3 up here, they happen inside the cell's RER. Right? You can see this is showing you that this is happening in here. While steps 5 and 6 happen outside the cell. Step 4 was actually the process of sending it outside the cell. Since this is a protein that ends up outside the cell, it is synthesized by the RER ribosomes, not free ribosomes. That's a little technical thing you want to make sure you know. So what is step 1? Synthesis. Translation of collagen alpha chains, usually glycine XY. Right? And X and Y are proline and lysine. Glycine content best reflects collagen synthesis because collagen is 1/3 glycine. It's also 1/3 it's also going to be 1/3 proline and 1/3 lysine for the most part, but not necessarily. What's confirmed is glycine. X and Y can just be all X's or all Y's as well. But glycine is always going to be there, so 1/3 of collagen is glycine. Then you have steps 2 and 3, which are hydroxylation and glycosylation. And this is pretty much the only time glycosylation is something we do, that we want to do. So hydroxylation is, just adding hydroxyl groups to proline and lysine, like you see over here. Glycosylation occurs only for the lysine, right? So you get your glycosylation of the pro-alpha chain hydroxylysine. It's already been hydroxylated. And all of this helps you get your triple helix of 3 collagen alpha chains. Problems forming a triple helix will lead to osteogenesis imperfecta, whereas hydroxylation is not possible if you have vitamin C deficiency. That's known as scurvy. Now, there was a question where on UWorld, they were telling you that they found some kind of material. They're trying to figure out where it came from. What would you need to find on this material to make sure you know it's collagen, right? And a lot of people chose glycine. But finding glycine on something doesn't tell you that it is collagen. In fact, if you're looking at something and it's 1/3 glycine, you still don't know for sure that it must be collagen. It could be something else that has 1/3 of it being glycine. But if you find hydroxyproline or hydroxyglycosylated lysine, then you know that you are looking at collagen, because nothing else will have hydroxyproline or hydroxyglycosylated lysine in a human. Right? So that's what this is all about right here. Step 4 is just exocytosis. So I mentioned your trafficking proteins here, right? You got your COP2. The pro-collagen is transported from the RER to the cis Golgi, right? So from the RER to the cis Golgi, that is the one thing that COP2 does. And then your exocytosis from the trans Golgi will use clathrin, right? So there's no COP1 activity. You're not doing any back to square one. Nothing is going back to the nucleus. So you do exocytosis, you send your triple helix pro-collagen outside. Outside, you have proteolytic processing, cleavage of the disulfide-rich terminal regions of the pro-collagen, and that gives you insoluble tropocollagen. You make insoluble tropocollagen. So you got rid of the C and N terminals, and we talked about that a little earlier. Right, so got rid of that. Problems with this cleavage can lead to Ehlers-Danlos syndrome because without removing the C and N terminals, it's not really going to connect very well when you try to do step 6, which is the cross-linking. Cross-linking is reinforcement of many staggered tropocollagen molecules by covalent lysine hydroxylysine cross-linkage, right? And this requires copper and lysyl oxidase to make collagen fibrils. Problems with this cross-linking are also related to Ehlers-Danlos as well as Menkes disease. And look at this question. This is from Amboss. 31-year-old man comes to the emergency department for acute tearing chest pain that radiates to the back. Despite appropriate therapy, the patient dies. Autopsy shows an increase in mucoid extracellular matrix and loss of smooth muscle cell nuclei in the media of large arteries. Which of the following additional findings is most likely in this patient? Acute tearing chest pain with radiation to the back is consistent with aortic dissection, a life-threatening condition. Mucoid extracellular matrix accumulation and loss of smooth muscle cell nuclei from cystic medial degeneration of the large arteries predisposes people to aortic dissection, and in young patients is indicative of connective tissue disorders such as Marfan syndrome. Deformity of the chest wall, such as pectus carinatum or pectus excavatum, is a common finding in patients with Marfan syndrome. And that was the answer for this question. You would find pectus carinatum. This condition is strongly associated with aortic pathology, including aortic dissection, aortic valve regurgitation, aortic aneurysm, and aortic root dilation. Marfan syndrome is caused by mutation in fibrillin 1 gene, which encodes for an extracellular glycoprotein. Now, Marfan is coming up, but I do want to point something out. In Marfan syndrome, right, you can see either one of these, pectus carinatum and excavatum. However, carinatum is more specific. If you see someone with carinatum, you know they have Marfan. Whereas excavatum, you pretty much still do know they have Marfan, but not 100%. Or at least not, carinatum is more specific than excavatum is. So look at osteogenesis imperfecta for now. Osteogenesis imperfecta is almost always going to be a giveaway. Like, you'll know this patient has osteogenesis imperfecta, you'll know this is OI. It's really hard to confuse OI with anything else. It's a genetic bone disorder, right? That's the first thing you want to remember. It's a bone disorder caused by a variety of gene defects, most commonly COL1A1 and 1A2. Most common form is autosomal dominant with decreased production of otherwise normal type 1 collagen. Manifestations can include multiple fractures with bone deformities, even though there was minimal trauma, blue sclerae due to the translucent connective tissue over the choroidal veins, and remember, type 1 collagen was on the cornea, right? So that's how you can remember that. Type 1 collagen is on the cornea, and everyone always remembers this blue sclera thing, right? Type 2 collagen was the vitreous body of the eye. Alright. So you can also think of it as vitreous body, or if you want. Then you have some forms have tooth abnormalities, including opalescent teeth that wear easily due to lack of dentin, and hearing loss because you can affect the ossicles, which are also bones. Diagnosis of OI is made by a skin biopsy to determine the structure and quality of type 1 collagen. DNA testing can also confirm, but DNA testing does not exclude because not all mutations of OI are known. There are some mutations of OI that we don't know. And, of course, there's no cure. So type 1 collagen is the predominant collagen in osteoid, the organic portion of the bone matrix. In OI, there's decreased osteoid production by osteoblasts. And you just got to make sure it's not child abuse. If it's about an infant who has broken bones, got to make sure it's not shaken baby syndrome. You got to make sure the structure, where the bones are broken, how they broke, what's it look like, stuff like that. Ehlers-Danlos syndrome. So faulty collagen synthesis causing hyperextensible skin, hypermobile joints, and tendency to bleed. And remember, Ehlers-Danlos is because you couldn't do cleavage of the N and C terminus. You couldn't do cleavage of the N and C terminus. And it can also be due to lysyl oxidase stuff, right? Copper deficiency. Right? Cross-linking down here. Ehlers-Danlos. Could be due to any one of these issues that happen outside. Now, multiple types. And you don't need to know all types. You don't need to be able to identify everything. Inheritance and severity vary. Can be autosomal dominant or recessive. And you don't need to know that either. May be associated with joint dislocation, bury in aortic aneurysms, and organic rupture. The hypermobility type with joint instability is the most common type. That's kind of what you're seeing in these images. The classical type, with joint and skin symptoms caused by a mutation in type 5 collagen. Vascular type, with fragile tissues including vessels, muscle, and organs that are prone to rupture, is type 3 collagen. So you really just need to know 5 and 3, right? If the question is just about joint and skin symptoms, it's type 5. But if it's about fragile tissues including blood vessels rupturing, type 3. Right? Last thing on this page is Menkes disease, and Menkes was also mentioned right here, Menkes disease, with the issue of copper lysyl oxidase, right? X-linked recessive connective tissue disease caused by impaired copper absorption and transport due to defective Menkes protein leads to decreased activity of lysyl oxidase, and that causes defective collagen. Results in brittle, kinky hair, growth retardation, and hypotonia. Now, for Menkes disease, just remember you don't see anything else. Always look for what you don't see in the patient, right? This patient doesn't have fractures. This patient doesn't have the cornea thing, so it's not OI. Right? They're not hyperextensible per se, so it's not Ehlers-Danlos. They don't have any rupture of organs, definitely not Ehlers-Danlos, so it must be Menkes. They don't necessarily have to mention the kinky hair. They can also just give you a picture of hair that definitely looks like, it's all coiled up weird. And this is going to present only in infants. It's not, Menkes disease is almost never going to be an adult popping up with Menkes disease. Alright? You can just go ahead and do these questions whenever you feel like. It's not, they're not really that good of questions to begin with. They're really very basic questions, but that's kind of what USMLE RX and Captain sort of do. Oftentimes when they're teaching you, they just give you really simple, straightforward questions that you will only get wrong if you haven't actually read the topic yet. But step 1 will probably never have a question of this type. Last thing on this for your molecular stuff is elastin, right? So elastin is made of tropoelastin and fibrillin, right? Tropoelastin and fibrillin. And it always looks like this, all messed up. But when you stretch it, you see it's able to stretch. That's why it's all messed up. So this is stretchy protein within skin, lungs, large arteries, elastic ligaments, vocal cords, ligamenta flava, everywhere. All of these specific areas. The lungs and the vocal cords are specifically very important. Rich in non-hydroxylated proline, glycine, and lysine residues. So you see how finding a lot of glycine in something doesn't confirm that it's collagen. It could be elastin as well. But the hydroxylated residues are only found in collagen. Tropoelastin with fibrillin scaffolding, and that's kind of what I was telling you right here. Cross-linking takes place extracellularly, gives elastin its elastic properties, broken down by elastase, and elastase is inhibited by alpha-1 antitrypsin. So if you have alpha-1 antitrypsin deficiency, your elastase is not going to be stopped. You're going to have a lot of elastase activity, which will go ahead and kill all your elastin, leading to emphysema and many other issues. Remember, alpha-1 antitrypsin deficiency is also related to the liver. Changes with aging are going to be decreased dermal collagen and elastin, decreased synthesis of collagen fibrils, and cross-linking will remain normal. Right? So the only thing that remains normal is cross-linking. They're going to stay cross-linked, but they're not going to be as many of them there. Marfan syndrome is an autosomal dominant connective tissue disorder affecting skeleton, heart, and eyes. It's an issue with fibrillin. Only the fibrillin, not all of elastin. Just the fibrillin, which is actually the scaffold, right? The fibrillin scaffolding. That's what the problem is with in Marfan syndrome. So your findings are going to be a guy, anyone, a guy or a girl, with tall, who is tall with long extremities, pectus carinatum, hypermobile joints, long tapering fingers, toes, cystic medial necrosis of the aorta. Remember, cystic medial necrosis is Marfan. Right? This terminology is very important. This is Marfan, cystic medial necrosis, and aortic incompetence and dissecting aortic aneurysms, as well as mitral valve prolapse. There's a subluxation of the lenses, which is typically upward and temporal. Right? So it's fanning out. In homocystinuria, it's actually going down. The lens dislocates downwards. Right? But this one over here, you can see the lens is upwards, looking at a fan, or it's fanning outwards because this is Marfan. This over here is a CT of a thorax in a man with Marfan syndrome, and you can see the large aortic sorry, large aneurysm of the ascending aorta, which is with the red overlay. You can see, let me make it bigger. You can see right there, there's an aneurysm, right? That's not supposed to be there. This is an image from Amboss. Additionally, a small aortic dissection, which is green. Oh yeah, this is the aortic dissection right here, right? In your ascending, this is the aortic dissection. And aortic dissection, you have to remember, there's multiple things. There's either hypertension or smoking that causes an issue. Aortic dissection is due to hypertension, right? The left main bronchus and right pulmonary artery are compressed but not occluded, and that's what's labeled in this image as well. So 1 and 2. So I like that Amboss does that. They kind of like color lay CTs and X-rays and really try to help you see everything in the lungs, especially X-rays of the lungs. They do a great job with. I've included all of that in this PDF as well. So yeah, that's it for that. Next topic are lab techniques. But what I am going to go do now is I'm actually going to go get some food. I'm going to eat. It's 3 p.m. for me right here, right now. I'm just going to continue on after eating. I'm going to take a little break, and I'll continue on at 5 p.m. So I'll be back in 2 hours to go on and keep going in biochemistry. So hopefully so far so good, and I'll catch you guys in a little bit. Thank you.